protozoa. So, in the right here, there is a tuberculosis incidence per 1,000 children. If you see, Fortaleza in South Africa become it's considered low incidence compared to the other sites, which was good to see this. But in Fortaleza also there is consistent to see Shigella here, which is very interesting to see Shigella is first, but then virus again, sapovirus, norovirus second, sapovirus third, and adenovirus, and then Campi over here. So the reason that I'm going to show you this is because we also explore these enteric pathogen in the semi-arid area in Brazil, which is showing this slide, the right part. You see 6 cities which we approach this. We have consistent here with virus, especially in terms of incidence. Since this is another approach, was Luminex was not the TAC-man, so Shigella cannot appear there as the first, but we have consistent with virus as the biggest incidence, and we do have this consistent with the TAC-man here seen in the urban area in Fortaleza. So the first time that we have data collected from the huge area in Brazil outside of Fortaleza, and it's very important for us for the public health. So recently, I'm like to show just this slide to show the approach of clinical metagenomics, which is increased accuracy and affordable to detect enteric bacterial pathogen in situ. If you see these Peterson et al. Study published in Microorganisms, they approach comparing metagenomic study as exam compared with the routine culture to check the sensitivity and specificity for Campylobacter, Salmonella, and Shigella, and then the result of that is a very excellent sensitivity and specificity of metagenomic approach to detect these enteric pathogen. It's the only study that approach metagenomic right now that I could search for the PubMed, but it's showing that it's possible. The paper mentioned that the proportion for the enteric pathogen regard to the other microbiome detection in this approach is very low. I mean, the Campi Salmonella is less than 2% of the total proportion for the other microorganisms searched in this metagenomic approach. But they show us that it's possible. I think the field is still in development. I think the paper showed how difficult it was to extract the DNA from bacteria and microbiome and take out the DNA from the host. But the method work. I mean, in other words, it's a very challenging field to start to think right now. So this is probably the best approach right now that I think needs to be done, not for the bacteria here, but for the virus especially, and expand this for the bacteria that has been also important in these enteric infection that I just showed you. So in summary for this first slide, I need to check my time. Can you tell me how much I have, Alexandre?
Yes, we probably started a little bit later, so you probably around close to 20 minutes.
Great. So that's me. Let's see if I can move quick. So the summary of this, it's very quick here. For the molecular diagnosis, we improved from 40 to 65% of the sensitivity to detect enteric pathogen. This is good. implement metagenomics. It's probably a broad view better for the co-infection associated with the disease that we're talking today. We see that the sapovirus was the predominant virus, which has not been detected before. Shigella actually was very surprised because with the sensitivity increase, the detection without actually having the blood diarrhea, which is very important to tell you here. And for sure, this method will increase the sensitivity and specificity. So the summary here also is, this is, I told you before, I mean, Shigellose, actually Shigella association without blood stool is no longer a good approach for having that etiology to treat with antibiotic. I think that's a message here. And this program going to show model for association with growth deficit. Here in the malady, enteroaggregative E. coli and Giardia was the one that has been associated with growth deficit. Diarrhea and growth deficit has been associated with Shigella, camp, and norovirus. And primary association with diarrhea, as you see, most virus and also enterotoxigenic E. coli. This is the information that I can see. Not all pediatrics in the developing count and probably in developed count has been using as an approach to understand and to diagnose enteric infection and getting important in terms of public health. But I think we need to review, we need to divulgate this in more specific clinical approach for the public health. So the second paper, it's also quickly, but it's important to show that now we touch base the subclinical specimen collect. So that's not enteric. That's most disease. I can see that most of this, when you see the inflammatory biomarkers, it's all associated with these children having these enteric pathogen detect, which means that these children has the environmental enteric dysfunction disease, which we're going to call this approach right now. So we see all pathogen. You see total here, just to mention, there is a decrease in lymphoid score. But as you see here, bacteria is more important and protozoa than virus, which not get the decrease in this lymphoid score. So if you see more specific, you can say that by 2 years, Shigella is an important causality for this. Enteroaggregative E. coli also, Campylobacter, Jejunum coli, norovirus, and Giardia here. It's very, this information is very important. So having these enteric pathogen present in the subclinical specimen can be a danger thing in terms of children development and growth development. And this has been also associated with the neurocognition development. As we done decades ago, yes, I think we, malady actually replicate what we've been seeing before. So, and to pay attention a little more for co-infection, I had Virginia talk this morning about the update of TAC-MEN and enteroaggregative associated with 3 or more pathogens, showing more sensitive of the TAC-MEN, showing this result that I'm just going to show this slide. As you see, if you see in red here, you can see enteroaggregative and 1 pathogen get decreased change in weight for length disease score. There is also change for weight for a z-score, seeing that I'm not going to show here, but when you have 3 or more pathogens associated with enteroaggregative E. coli, you have a significant decrease on the weight for length disease score. And surprise, when you see 3 or more, you see 3 or more, 1 or more enteric pathogen without Escherichia coli enteroaggregativa, we cannot see these decrease. That means that these E. coli has some impact in terms of co-infection with other pathogens. Missing here, and that's the willing that we'd like to do, is the metagenomic of these stool sample to approach this not only from the co-infection of enteric pathogen, but also with the microbiome microorganism. So next is a summary of that. We show you not seeing this. This is changing my reading here, but let me see if I can see. Subclinical infection, né? And pathogen count, particularly Shigella enteroaggregativa coli, Kempenchiaria, had that substantial negative association with linear growth. And of course, successfully reducing exposure to certain pathogens can reduce global stunting, as we show in this paper before. This is a subset of the co-infection just with this enteric pathogen EHEC. You can see that there is a negative association with delta, weight for length, and also weight for Z score. So, and we have new data that actually Livia showed quickly today, associated with the new method that has been used for the lactulose mannitol and barrier function, a non-invasive method. As you can see, Fortaleza here, recent, we have done a case control study, pick up children from the same generic similar population of Fortaleza. As you see, Fortaleza is a poor city. The human development index, it's already here, it's very low. And the few area here in green is the only area that we could get some children or, with the same genetic background that we can compare with the control children collecting in the case control in Malad and case malnutrition collecting here to do the lactulose mannitol test. And the exploration here is to ask the question about if you have a similar population, genetic population, if you have differential approach to see intestinal barrier function in children in a health condition, in the economically good condition in Fortaleza compared to controlled children living in the peri-urban area and also to malnutrition children. That's the biggest issue that this data is showing. So this data is not published, it's going to be sent for publication. And here is the result. If you see the excretion of lactulose, you can see that there is a decrease of that excretion on malnutrition, and also in the control that we call it from that case control as environmental enteric dysfunction. As you see, both get decreased compared to the control. When you see the mannitol, we can see that the control over in the economic area, good economic area, we have an increase of excretion of mannitol compared to the other two groups. And the lactulose mannitol ratio, as you see, there is a very decrease on the control in good economic situation compared to the other two groups. That means that lactulose mannitol non-invasive test can really detect the decrease in area of absorption and also detect decrease in lactulose, which means for me that when you decrease the villus area, you also decrease the permeation in the tight channels for lactulose. That's the reason you see also decrease in the lactulose excretion using this test. So there's very consistent data for me. It's consistent with the animal data that Samili showed this morning. And it's very interesting to have that data on hand. So, and right now you can see that the enteric environmental enteric dysfunction, malnutrition has a huge pathobiology, subclinical and clinical, which can reach easily the malnutrition and increase the vicious cycle. I mean, there is data now from the group that's showing several things. We showed this before: neurocognition decrease, physical development, Z-score decrease, we have this ratio actually decrease, and we also have these inflammatory pro-cytokine and chemokine increase, which is associated with these changing the intestinal barrier function, and also biomarkers here that can be well used to parameter to follow up this disease. So it's a very detailed pathobiology that's better than before when we showed that slide, that we now can understand a little bit more. So, and this paper that I just found get this gut microbiome association with the intestinal inflammation and permeability and damage in the young Malawi children. I think it's very interesting because they use here the maturity age for age Z-score in this microbiota metagenomic analysis, as you see here, and also the Shannon index, which measure the diversity of these microbiota. As you can see, there is always an inverse correlation when you see calprotectin. As you're getting more mature, you get a less calprotectin, which means that less gut function damage. Also seen with alpha-1 trypsin and also reg-1b, which also measure damage for the intestinal barrier function. And here we have also the same thing, showing that there is, if you have a more diversity, you have a less risk to get these biomarker, which we get a less risk for the environmental enteric dysfunction. So this is a very interesting data. I think this is very stimulating to approach metagenomics. So, and now because of the time, I'm just to show you that the microbiome has been associated with the malnutrition and also with health children in this cohort, including MALAD. As you see here, just to show the centroid in red here, you see the health children. The centroid is completely different from the centroid seen in the severe acute malnutrition children over here. So, and there is some probiotic which can be changed, at least from the moderate acute malnutrition to normal health. These microbiota here, which has been associated with these health children, and also diverse, less mature microbiota associated with the malnutrition. So, conclusion, finally, I think my time is probably too much. Yeah.
You're right.
It's very simple. It's very simple. These results demonstrate a multiple presence of microorganisms associated with environmental enteric dysfunction, enteric infection, malnutrition in children, with a consequent death in growth development. And this is probably stimulate us to think more about this approach of metagenomics. Thank you very much. And also I'd like to show all of these people. Brazil, essentially at the University of Virginia. Here is Dick over here behind. Here is the MALAD group, only call for the principal investigator and major collaboration, which is a huge group. And that's, we miss very much this group. And let me finish it. Thank you very much.
Thank you, Aldo. And we are open for some questions.
Hi, Aldo. Great talk. Thanks.
Yeah.
Has anyone used the sequencing approach to look at how the gut microbiome might protect against different pathogens or kind of even increase susceptibility to different pathogens?
The paper, Jonathan, which I'm going to call back here quickly. I think I took out the screen. Yeah.
Yes.
Yeah.
Back. Yeah. It's quickly. I can show you one good thing. It's getting to you now.
Yeah, I can see it. Yep.
Okay. This paper is very interesting because when we let me increase here. Yeah. When you look for these results here, I mean, all of these 15 microorganisms isolate from the metagenomic microbiota has been associated well with the health children. Means that this healthy microbiota protects against these environmental intact dysfunction. It's not causality yet proved because there is no cohort to show that, but it has been shown in Bangladesh and also in Malawi and some other children. And when you see the severe acute malnutrition children, the centroid, we get an immature microbiota compared to this one, and also the diversity of these microorganisms is less than this one that is showing for the health children. So the good hypothesis, yes, there is this good chance that this microbiota can be, and actually Jeff Gardner has been isolate 5 of these most invasive healthy microbiota, which not necessarily all 5 will protect against the immature microbiota, but actually by a few, 2 of them can replace the bad microbiota and prevent the mice to get to the malnutrition final point. I mean, in other words, I think it's like a postulate. I mean, I'm very impressive about this paper. Actually, there is two other papers that has been shown this line. That's very interesting, and also consistent data, and also I think I do have all of these 5 most invasive microbiota that has been isolated. I do have in my lab. This is a postdoc fellowship for the Samili, who is going to finish the PhD right now. And with the new diet, I think all of this, it's going to be dynamite to explore, not only here, but in Virginia and also at your site, Jonathan.
Yeah, lovely. Great, thanks.
Any more questions? I can hear
Hi,
some.
I do have a question. Dr. Aldo, thank you for your talk. Really nice. So my question is kind of related to Jonathan's question, I think, but maybe in a different perspective. So you beautifully showed the malady data showing that maybe specific pathogens could be related to EED. And then you showed some Jeff Gordon's data showing that the microbiome could be related to EED. And then I wonder if it's better to restore the microbiome maturity than focusing on a specific pathogen as Shigella or some viruses. Can you expand on that? Do you guess if it's better to focus on microbiome and in a general approach, or is it better to focus on a specific pathogen?
Yeah, that's a good question. I think the whole approach for this talk for me is to, as far as I understand, not only microbiome, but also metagenomic, including enteric pathogen. And when I say enteric pathogen, which including also bacterial virus, especially, and also a couple protozoa. I mean, I think we do have our approach here is going to develop this. We're just getting some support for the Illumina sequencing and also for proposal that I hope to have some support to develop at least like we have shown quickly in the PubMed a few results that's also positive, stimulating that type of approach. Now, Dick, it's like, I think for me, it's like to have another malad doing some metagenomic. It's crazy, but what happens with us? I think we had 8 sites, and the only site that I know is still with sample in the refrigerator for the whole time is Fortaleza. I mean, I just asked Melinda Gates Foundation to say, oh, we have this, we need to do some preliminary work in metagenomic. I did ask for the Illumina machine, but I already got, so I don't need the machine, but probably that's one good approach to have a second one. But in other words, the only site capable to rerun metagenomic is Fortaleza. And I think we do have some potential preliminary metagenomic. Pedro, the answer is not only microbiome, but metagenomic, including enteric pathogen. Even though it's a proportion is small, as you see, the paper that I showed you already detect with a high sensitivity and specificity, which means that's possible.
Hello, I just wanted to make a very quick comment to Jonathan's important question about sequencing as well. As Jonathan knows very well, there is a lot coming on with things like C. difficile and specific microbiota that may be helpful. There's a lot of interest in that. There's a need for that. And that kind of work hopefully can direct where we go from here. I agree with everything you're saying, and I know Pedro and I have talked about some things too, so it's exciting. Thanks.
Okay. Thank you, Dick. Yeah.
Can we move on then?
I think so. Yeah.
Hi, Sean. So,
Hi, Alex. Hi, everyone. Sorry for joining late. And Aldo, fantastic talk.
Yeah, thank you, Sean.
so Aldo, are you taking the lead now? Since it's myself. You're muted.
Go on for one more and then I'll take challenge. Yeah.
It's me.
It's you. Oh, thank you. Let me escape my line, my sharing here. Sorry. Let's see. Stop share. Stop here.
Wait.
Yeah, thank you. Yeah, you can start sharing your screen and I introduce you. Yeah.
Oh, actually, let me just. I'm not able. Just 1 second. I'm not seeing. No, sorry. I thought that the PowerPoint was already on over here and I cannot see, sorry.
Yeah, let me introduce Alex.
Thank you. Just to try to
Yeah.
introduce while I'm looking for the PowerPoint.
My pleasure to introduce Professor Alexandre Havit, who has been working here with all of these malad tools, TACMan array, and also now we are in virulence traits for the Enteroaggregative E. coli plus. We now explore more genomic collaborative work with the CAPS print here in outside, and also is going to be inside. But what I'd like to say is, Alex find good, interesting discussion about virulence of Enteroaggregative E. coli and diagnosis of this bacteria. It's intriguing but important, as we show data before, that this bacteria itself co-infection with other cause the death in growth development. And we do think that there is more virulence trait to understand, and also for not only understand, but also for diagnose itself. I think Alex has been a great help here with us and doing good work on Maled already. So I hope he can now start. Thank you.
Thank you, Aldo. Sorry, at the beginning I was not fine the PowerPoint slides, but then it's interesting, and I'm really pleased to be here talking about this work. And then my lecture will be entitled Redefining Integrative E. coli, a Genomic New Perspective. And what I'm going to talk about today is the data published by the group of Nataro and the collaborators of him from Denmark, who's Nadia Boysen, who I was pleased to meet when I was in meeting Nataro a long time ago. But it actually is a good perspective, since I'm not going to show any data from us, but showing the data from Nataro. But our perspective about this work is because we are already doing kind of exactly what they're doing with this paper published in 2020 with our collaborator in Germany, since we are already doing some sequencing of the EAG samples from our side. So it was a very interesting perspective and showing what they already find, and take this opportunity for emphasizing what they did and what we can do in the near future. So a little bit of some of history about the EAG, the integrative E. coli. This bacteria was first described by Nataro in 1987. This bacteria at the time had a different adherence pattern from EPAC, intrapathogenic E. coli, and it could not produce any ETAC enterotoxins, but it did have a stack break appearance when they perform HEAP2 cells assay for the adherence. So it was exactly a different bacteria from EPAC or ETAC. So it was originally identified in a child with diarrhea in Peru. But in 1995, Nataro also did published another data about volunteers who got the 042 strain, and they realized that that strain at that time could cause diarrhea. A paper published by Okek in 2000 also presented that these EAG could cause acute diarrhea and persistent diarrhea. And later on, Rogowski in 2017, taking use of the data from Maled, already also figured out that EAG could cause linear growth faltering among children from low-income countries. But not always EAG causes symptomatic illness. It always can be asymptomatic too. But this particular bacteria is more interesting, since it can be really genomic plastic. What is that? It's that what we saw in the German outbreak. This EAG, this German outbreak in 2011, they found that the cause of that outbreak was an EAG bacteria producing Shiga toxin. And then it was really interesting to see how plastic this bacteria could get. In the perspective of pathogenesis, EAG can cause mucosal inflammation, epithelial damage. It can get affinity with any segments of the intestine, and its pathogenesis steps, it's related to adherence, production of cytotoxins and enterotoxins, and then cause mucosal inflammation. But this EAG has been always famous about being really heterogeneous bacteria, especially about the array of virulence factors. And the most important one that has been detected is the transcription activator encoded by the gene AGGR. Many studies, many research already being classified the EAC when they have it genetically, the AGGR, as the typical EAC. And this gene can control like 44 chromosomal or plasmid genes. And among those genes that the AGGR can control, we can see all the 5 types of aggregative adherence fimbria. It also controls its proper repressor, encoded by the gene AAR, and also control the dispersine, which is a protein encoded by the gene AAP, and it detaches the EAG from the cell in order to contaminate another cell. But the point about EAC is that no single factor has been consistently implicated in EAC diarrhea. And also the other authors, other researchers already has been thinking about what is that EAC, what is that bacteria that do not present this transcriptor activated encoded by the gene AGGR, but as a typical EAC, but it's able to adhere to HEP2 cells. So it was this point that the people from this work, from Nataro, got interested to figure it out what it was going on with EAC, performing a whole genome sequencing study. So, sorry, why is it not going further? Okay. So what was the study design? So the EACs that they sequenced in this study was from the Global Enteric Multicenter study, a study called JAMS. It was a prospective matched case control study, and they enrolled children aged from 0 to 59 months, and they enrolled children who could have moderate to severe diarrhea, a kind of different from what we got from MALED studies. And this study was set in Africa and Asia countries. And in JAMS, they took 97 EAC strains, they found it, and there were 57 cases and 40 controls. In all those 97 EAC strains that they isolated, they were from Bangladesh, Mali, and Mozambique. At the time when JAMS was running, EAC was diagnosed by the genes AATA and AAIC. And at the time too, they also performed PCRs to characterize the 5 kinds of fimbria that they already knew that EAC could produce. But they wanted to really know if there was any other in this set of strains that could not present any fimbria genetically. And all these 97 EAC strains were compared against other E. coli strains, not all EAC, but also other species of pathotypes of E. coli. So the DNA, the genomic DNA was isolated by a kit specifically already from QIAGEN. The library for sequencing was prepared by Nextera using the reads from 150 to 250 base pairs. And the sequencing was done by the Illumina machine, and all the raw data was deposited in the European Nucleotide Archive. In order to perform the analysis, they used 5 different databases included, found in CGE. So they figured out the multi-local sequence typing. They performed the MLST using the MLST finder, using these 7 losses to figure it out about the 97. They serotyped these bacteria, these 97 strains, using the serotype finding, and also used the virulence finder. It's another database found in CGE. And they used the same cutoff about the data they found, 85 sequence identity and 60% of sequence coverage. They also perform phenotyping using phenotyper, and they also predicted antibiotic resistance using ResFinder. So they use all these databases to analyze the data they found about those 97. So they also perform phylogenetic analysis using a in silico genotyper, especially based in the SNPs, in the single nucleotide polymorphisms. So they took 9 EX reference genomes, and they also use other E. coli and Shigella genomes to perform this comparison. In order to verify the phylogeny, they use a software I told. They also did a large-scale blast score ratio. They used the predicted protein coding by Protigol, and they also perform a Fisher exact test to compare the virulence genes among cases and controls. I think someone is talking outside, so please shut the audio, please. Then the results, what we found, the most interesting results about the genes that were regulated by AGGR. So they perform analysis using all the virulence finder analysis for all the EAC known, the diarrheogenic E. coli, and also about extraintestinal E. coli too. And the most interesting find about the genes regulated by AGGR, they were all exclusive for EAC, as we can see by the pathotype species. But the most interesting thing that they found over here is that before this paper, they were using the gene AAIC and the gene AATA to diagnose EAC. But from this 97 isolated EACs, it was interesting that they found only 38% of these 97 samples positive for AAIC, a gene that was a chromosome. It is a chromosomal gene, and it was used for diagnosing. So it was really interesting to find that. AAP was the most frequent one, and it's always like that for many other papers published for EAC. The presence of AAP is really high. But the most interesting thing was seeing that AAIC was really low prevalence, and AGGR was presenting exactly most the same prevalence that we have been finding in many other papers, okay? Among the genes that were not exclusive from EAG, we can see a bunch of genes. They figured out, they analyzed like 200 virulence genes of all those E. coli. But it was interesting to find that 2 genes, they were specific for extraintestinal E. coli and avian E. coli, that those 2 genes, ESS and LMT, they were associated with children with cases, with children with moderate and severe diarrhea. Another result that they also found, they analyzed the whole set of toxins produced also by EAC, like all those 5 autotransporters toxins related to EAC and related also to other kinds of EAC, diarrheogenic E. coli. But the most interesting about these children who were enrolled for moderate to severe diarrhea was that again showing the same data from other papers, that the toxin SAP-A, which is a Shigella extracellular protease, was also showing association with the cases in this study. And it's interesting to see that almost the majority of those samples that were showing the SAP-A was also presenting the Fimbria type of Fimbria number 4. So 58% of them were showing SEP-A and Fimbria 4. Then the authors also presented a very good analysis about the Fimbria. So they found exactly that 73% of the strains were showing at least one of those Fimbrias, the known Fimbrias, the one from 1 to 5. And this Fimbria is encoded by a 4-gene cluster. It's a gene cluster presented by an usher, a chaperone, and 2 palins, a major and a minor palin, okay? And it's represented by all 4 of genes as I'm showing over here. So AGGD is the chaperone, AGGC is the usher, and B and A is responsible for the minor and encoding minor and palin and major palin. So among the data they found about the prevalence of these Fimbria, the most found was the Fimbria number 1, like 22%. But there's also another structure difference between all 5 Fimbrias already known, is that the only one that is kind of different is the Fimbria number 2, because it is not distinctive by just one cluster, but it has 2 distinct clusters. I'm going to show you a picture right in some minutes, and then you'll figure it out. What's the difference between all these 5 Fimbrias? But the Fimbria 2 has a distinct structure distinction about the other 4. So, and this paper also was most interested in the ones that did not show any of these known Fimbria. So they found 19 strains that were negative for these Fimbrias, or these 5 Fimbrias. It did not adhere as an aggregative pattern. From those 19, 3 of them adhere to cells, but not the way as an aggregative pattern. And 1 of them was a cytotoxic. But the most interesting about these 19 strains is that 10 of them were AAIC positive, that gene that it was used for the diagnosis. But 9 of them were negative for AAIC. From those who had AIC, 8 were negative for any encoded genes and also negative for AGGR. But 2 of them were positive for AGGR and the genes that it's normally regulated by AGGR. From those 9 that were not AAIC negative, 6 of them were AGGR positive. But 3, the interesting thing about those 6 is that 3 of them were having the same virulence profile and all belong to this serotype O166 and H15. And the 3 last ones were negative for AGGR and harbored a small amount of genes that were regulated by AGGR, even though it did not have the AGGR. And here they also perform, as I told you that I was about to present this picture, it's a simplicity way of showing how type 1, 3, 4, and 5 Fimbria are similar in structure. We can see by here in this number 3, it's this number 3. So we can see for 1, 3, and 4, and 5, they cluster as a group of genes like the major palin, minor palin, usher, and chaperone. So they all are structured like this, showing the cluster here as I'm showing here. And I'm showing here, also here for 5, and also here for 5, for the 5th one. But the clusters are divided in the Fimbria too. We have some part of the genes that encode the Fimbria here and separated by the other ones too over here. So it's kind of distinct. But what they also found is that there was a kind of different, like 3 strains from those 97 that they were presenting a very different structure of Fimbria. When they took a close look at those 3 Fimbrias that were negative for the type of Fimbria they already know, they are the 5 already known from them, they were harboring AGGR, but they were finding a colonization factor peeling similar to the ones presented in ETAC, what we call the CS22. They also were presenting a chaperone and an usher similar to the ones that they have been finding in Salmonella. And they presented a minor peeling now with this gene presentation. So when they go a little bit further, analyzing this kind of cluster, the CS22, when they predicted the proteins of this factor peeling colonization, they figured out that 14% it was similar to an archetype of aggregative fimbria. But when they predicted the isoelectric point, the AAF fimbria, similar with the ones that already have been finding in YAC, had a PI as 8.0, but this CS22 encoded gene display a low predicted PI as a 6. So it could not be related as a novel aggregative peeling as it shows normally for EAC. When they sequence the whole plasmid of this, one of those three strains, the C67115, they found a bunch of putative EAC virulence factors. And when they also entered in an intro-based database and they looked for 469 strains that were already sequenced around the world, they found 5% of them positive for AGI, 5% of them, presented this gene cluster like the CS22. So because of that, they already figuring out that in saying that strains that has this CS22 like adenosine, it's a basic typical EAC. They also perform adherence test, but this time in a colonic organoid test, a little bit different from the HAP2 cell array normally done. And they figured out that the strain that has the CS22-like cluster gene presented the same adherence pattern as the 042, the one that has the fimbria number 2. But when they mutated this 042, it did not show. So both EACs, a fimbria noun number 2 and a fimbria kind of CS22, was showing the same pattern, the same intensity of adherence in this collonoid organoid. Another interesting about things that they found when they perform a neighbor joining tree analysis of all these 6 kinds of now fimbria, they found that the fimbria 1 type could be distinct like in 3 subclusters, which we didn't see this before. And the last analysis that they performed, the phylogenomic analysis of EAG, constructed using 234,000 conserved SNPs, they figured out that 42% of those 97 strains were belonging to the phylogroup B1, 29%, almost 30, belong to group A, and 23.7 belong to phylogroup D. And the interesting thing is that 68% of the ones that did not present a known fimbria were belong to group A. And they also figured out that the group D could be distinctive in separated in three lineages, 3 different lineages, 1, 2, and 3. And they also figured out that the D group number 2 over here. Now, one of those 3 find the CS22-like were phylogenomically analyzed here, now found here in the group D, and the other 2 were here said in group B1. So the most interesting discussion about this paper is that, of course, they reinforce the data that the importance of AGGR as a regulon in the classification of EAG as a typical EAG when they presented the EG and the AGGR. But the point about the challenge of performing the HAP2 assay, which is a really challenge and it takes a long time to do it, it was much easier for the studies to investigate the epidemiology of EAG, was also using the AGGR and the associated genes and the AGGR as the de facto standard way of analyzing the EAG epidemiology. And then the most interesting that this paper just brought to the whole researchers that used to work with the EAG is they brought a new molecular definition of EAG, saying that for now on, we could not use the AIC and the ATA genes for diagnosing EAG, but now the EAG molecular would be characterized by the presence of AGGR in a complete cluster of AAF encoding gene. They should have usher, chaperone, and both minor and major pilling, but also the ones who could have the CS22-like cluster. Another point that they discussed is about the association with the moderate and severe diarrhea. Now, they found something that they didn't never find before, that some genes that were not exclusive EAG genes also could perform and be associated with moderate and severe diarrhea. SAP-A was already a gene exclusive for EAG also, but they were already shown before in other papers that they could be associated with this intensity of diarrhea. But this is the first time they're finding genes that it's not regular find in a EAG that is showing some associated. The ESS and the OMT gene, they're all more associated with causing urinary infection. And they also, ESS, for example, is a complement resistant related gene. And OMT is a protamine inactivation. So they're both related to urovirulence infection. So given the fact that EAG has been already implicated as an uropathogenic bacteria, it is possible that both authentic enteric and urinary system pathogens can be found among strains meeting this new definition of EAG. Another point in his discussion is that AAF adzenes could be a promising immunogens for developing vaccines against EAG infection, since they are really high prevalence and high amino acid conservation. So as a conclusion, EAG remains a common bacteria associated with diarrhea and linear growth, but it has been also associated with extraintestinal disease. So as a perspective of this work and what we have been trying to do with our collaborators in Germany is that the natural history of EAG can be enriched by a global genomic analysis in order to verify if the new molecular definition presented here by the Netaros group in collaboration with Nadia in Denmark could be used worldwide. Thank you.
Okay, Alex, excellent. I think as we are seeing, there is a lot of work to be done again for the integrative association with the rare disease, environmental enteric dysfunction, and malnutrition. So what I'm seeing here is James started using genomic for approach to associate the genomic with the rare disease. Alex, just to start the question here, how do you see these genetic, the exploration of the genomic of enteroaggregative E. coli in malnutrition and also environmental enteric dysfunction? I think it's going to be another type of understanding for the bacterial virulence association. Do you think that it is the correct way to start to planning this?
Yeah, I was here, with you and Ataro when he came to as a visitor in one of those TMRCs, inclusive, that when he presented the use of AAIC and AETA as those two probes to use to diagnose EAC. So what they're presenting now for us is that most of the job that we have been saying that EAC was associated with malnutrition and associated with diarrhea in all those studies that we did not find any association with the EAC can be totally changed after this new definition. So it's another way, but what I can say is that this study was done with samples with moderate and severe diarrhea. So many things can be changed when we figured out what EAC is doing in children from ILAD that did not present diarrhea at all. So it's interesting about that's what I'm thinking. So I'm not sure if I was answering what you have asked.
So no, you're okay. I mean, I think that the probes for exploration metagenomic need to be rethinked in terms of how to approach better interogative E. coli. I think for diarrhea disease approach, it's the one that you just showed. But Dick is asking one good
about the
question
specific
here about
associated. Yeah,
Inflammation, sorry.
yeah, no problem, Dick. What the point was, the only 3 genes that they figured out that they have some points about associated with this diarrhea thing was those 3. And they did not perform any group associated. So they didn't put many genes together to see if there was a combination of genes in order to see if they could put it together, could be associated. So so if even though they probably used to figure out genes that were related to inflammation, they didn't find any associated after this analysis. So only those 3, and only one was exclusive for EAC, was this the SAP-A gene, which were already a toxin before and associated with diarrhea. So the other 2 were not even EAC genes exclusive. So that was the interesting part of that.
I remember well when Nataro was there in your lab with us at the time. And I really was, you may recall, I kept asking about agar because that's sort of the one that controls everything else almost, but not entirely. So we don't have agar data in the probes from LAD yet.
No.
You should get it.
What we did for the TAC, for diagnosing EAC, they didn't use the proper AGGR. They were detecting it, but they have data when they did the TAC analysis. AGGR, it was, in the TAC paper, they use, as I told you this morning, EAG, AAR, the negative repressor, and those 2 AAC and AATA. They changed. So the point about this change, the whole change, is also because of the way the analysis was done. We have a bunch of data, but figuring out, doing disassociation, doing a whole genome sequencing analysis, and figuring out this new definition changed the whole thing. So that's the importance about doing the whole genome sequencing. So before that, they were all using genes exclusive for EAC, and the ones that they were finding. Now they figured out that the EAC is not only an intestine pathogen, but it also being in the urinary tract. That's another point.
Sure. Yeah.
Sure. Yeah. Well, I think we have time is out. I mean, it's late, but I think, thank you, Alexandre. I think we're going to move for the next talk.
Thank you.
Is going to be Dr. Shamuri, going to talk about the bioacid profile reveals distinct signature in undernourished children with environmental enteric dysfunction. Dr. Sean, please.
Thank you, Aldo, and thanks to all the organizers of the TMRC for the chance to be with you today and share work that many of you inspired. I see Dick and Aldo, my mentors, and so this is extending from their work in South America to show some work from Pakistan. So let me jump into things here. So I want to start with by acknowledging a recent accolade that came to the lab because of two people you know very well. So this is Vinicius, who is spending time with us as a postdoc, and you'll recognize Desiani there. So both folks well known to EB Med, and so Vinicius and Daisy helped lead the recent certification of our laboratory as a green lab at UVA, and we just learned that we're the second laboratory to achieve gold status for environmentally responsible practices in conducting the laboratory, how we manage our cold chain, how we dispose of chemicals, our use of electricity in the lab, and so you know. I think that there's a delicious irony because I think people in the United States are used to lecturing Brazil about environmental issues, particularly with the Amazon. And so to have Vinny and Daisy lead our laboratory in becoming environmentally responsible was really wonderful to see. And I'm very proud of the accomplishment. So we celebrated that this week. So objectives for our study, I'm going to give you a brief overview of the study of environmental enteropathy and malnutrition in Pakistan. Or SEAM Pakistan. I'll show some serum and duodenal bile acid profiles from SEAM and their correlations with childhood growth, biomarkers of enteropathy, and gut histology. And I'll tell you ahead of time that the key findings are we saw a lot of subclinical liver dysfunction, cholestasis in these children, as well as decreased secondary bile acids. And these were associated with growth failure, inflammation, and also enteropathy severity as measured on biopsies in a modest number of kids who underwent endoscopy because their malnutrition did not respond to nutritional interventions. And then I'll spend a little time talking about potential implications of our findings for reversing the vicious cycle, what that might mean for nutritional rehabilitation, fat malabsorption, and also growth hormone resistance. So the conceptual framework for SEAM was, I can say, can be summarized in this idea of tissue is the issue. So I'm a pediatric gastroenterologist, and one of the common modalities for in high-income countries for evaluating children who are having difficulty growing and also who have enteric disease is to actually access gut tissue. And so we asked the questions where using biopsy-based approaches nested it within larger cohort studies, similar to MALI-D, what could we learn about phenotype, host genes, and microbial community configuration, and how that plays into environmental enteric dysfunction. And so this approach allowed us to ask a lot of questions about sort of how clinical and pathological phenotypes match up. What does the microbiome look like, not just in the stool of children with environmental enteropathy, but look like actually in the upper digestive tract? What is the transcriptome of the duodenum, children with this condition? And then one area that I'll be focusing on in this talk are really detailed bile acid profiling. So we know that bile acids are a way that the liver and the gut communicate, and that also the bile acids act on the microbiome, and in turn the microbiome acts on bile acids. And so we thought that this might be a key to getting a greater understanding of the vicious cycle. So our studies were conducted in Motility, which is in the state of Sindh in the country of Pakistan. And so on the left here, we have Pakistan and the Arabian Sea. Karachi is a mega city in Pakistan and is the home of the Aga Khan University. And so this work was done with my collaborator in the field site led by Asad Ali, who many of you may know from Malidi. And then AKU's main field site, which was established by Zulfi Bhutta several decades ago, is in Motility, which is a rural area with a lot of sort of agricultural and monoculture of cotton. And so it's now, 1997 was my first trip to Fortaleza. And even back in the mid 90s, I don't, that those experiences did not prepare me for the degree of poverty and undernutrition that I have seen on my several trips now to Pakistan. And so here's just some geographical and demographic information about Motility. So 85% of the population lives in rural villages. Almost 30% of the population is severely stunted. And there's also 3% of the population is wasted. A very high rate of poverty, with over half of people living in poverty. But there is good access to improved sources of drinking water. And there is low breastfeeding rates. So only 3 out of 10 children are exclusively breastfed through the first year of life. And then only 9 out of 20 women of reproductive age are have adequate diets. So this is an underdeveloped area with a lot of undernutrition. And so I mentioned Asad Ali, who is in the red shirt here. And this is a visit that he took me on to one of the local villages involved in our study. Beautiful families, beautiful children who are, most of them are employed doing agricultural work for wealthy landowners. And so marginal diets and lots of exposures to enteropathogens. So we enrolled children between the ages of 0 to 3 months, and we were interested in finding children who were wasted or non-wasted, were recruited into the study, and then had weekly and monthly follow-up visits by community health workers, looking for morbidity and also measuring anthropometry. And at 3 to 6 months of age, and again at 9 months of age, stool, urine, and blood were collected to look for different biomarkers. And those children who were progressing to wasting, or excuse me, who were wasted as measured by wakeful length Z-scores, were given a twofold nutritional intervention. One was an educational intervention, and then one was daily provision of a supplementary food called achimum, which is chickpea based. And then after these nutritional interventions, children were reevaluated, and if they were growing poorly, they were offered evaluation by a pediatric gastroenterologist, who then decided whether or not an upper GI endoscopy might be indicated, sort of using European and North American guidelines. And then the treatments based on findings. And so, all right, so for the endoscopies, we obtained research biopsies from the second and third portion of the duodenum, and these were analyzed by RNA-seq and also had 16S sequencing of the biopsy and also aspirates from the duodenum. We controversially had a group of control children from Cincinnati, Ohio, and these were children with gastrointestinal complaints who were a couple years older than the average age of our Pakistan cohort. And then we also had children who were newly diagnosed with celiac disease. And so the old name for celiac disease is non-tropical sprue. And there's a lot of interesting overlap in the histopathology of celiac disease with environmental enteropathy. So we thought that that could provide an interesting disease control. And then we initially were enrolling some new diagnoses of young children with inflammatory bowel disease, but dropped that later in the study. So I'll jump right into the findings of the bile acid profiling at these two infantile ages, so 3 to 6 months, and again at 9 months. And these bile acid profiles were done by targeted MS mass spec in the laboratory of Ken Setchell at Cincinnati Children's, who has a particular expertise in bile acids and clinical mass spec for children with bile acid disorders. And so we had from our cohorts 339 undernourished children from the Montieri area, and then 50 healthy controls. And so Ken's group initially described what were just total serum bile acid concentrations. And he found a high concentration of serum bile acids in the undernourished group. And these were elevated enough to suggest what we call cholestasis, which is impaired flow of bile from the liver into the digestive tract. And so as bile accumulates in the liver, it enters more systemic circulation, leading to elevated serum bile acid concentrations. And then specifically, biomass spec looked at these different bile acid species. And so the two interesting ones that were elevated at both age groups were glycolic acid and glycochenodeoxycholic acid, which were seen elevated 3 to 6 months, and again at 9 months of age. And these were consistently more elevated in the serum of children who were undernourished. So looking further into that, we had done a number of other biomarkers in SEAM, and we had also analyzed serum for different inflammatory cytokines. And so here I'm showing correlations of glycolic acid and secondary bile acid concentrations with serum cytokines. So we saw from interferon all the way down to IL-12, significant positive correlations, and then an inverse correlation of secondary bile acids. I apologize for the jumpy mouse, I'll just use my keyboard, with secondary bile acids. And so secondary bile acids, as I mentioned earlier in the talk, that transformation of bile acids from primary bile acids into secondary bile acids occurs in the lumen of the gut, and that's done by the gut microbiota. And so our finding of decreased secondary bile acids in the serum of these children suggests a dysbiosis that was impairing the transformation of primary bile acids into secondary bile acids. And that was decreased presence of secondary bile acids was associated with a higher IL-1 beta signature in the blood of children. So in a group of 63 children who ultimately underwent endoscopy because their malnutrition was so severe and it did not respond appropriately to supplemental nutrition, we were able to look at the severity of their enteropathy in correlation with both their plasma serum bile acid profiles and also their duodenal bile acid profiles. And so we saw correlations of our plasma levels of glycolic acid with total EED severity and also PANA cell density. One of the features of the EED score is those children with a higher degree of environmental enteropathy seem to have less PANA cells present on their biopsies. And then secondary biopsies, we saw interesting correlations with epithelial detachment, the presence of intramucosal Brunner glands, and then also eosinophils. And then on the bottom here, we're looking at the correlation of both aspirate bile acid profiles and plasma bile acid profiles with more systemic biomarkers. And we found an interesting correlation of GCA and secondary bile acids with platelet counts. So platelet counts can serve as an acute phase reactant and gut inflammation. So we can see this in EED and also an inflammatory bowel disease in children, where sometimes an elevated platelet count is just a marker of systemic inflammation. So it was interesting to see GCA and secondary bile acids correlating with that. And also we see this interesting hit with circulating eosinophils as well, and plasma GCA content. So another question that we wanted to ask from this data was, because of the impracticality of doing upper endoscopies and collecting duodenal aspirates from children in low resource settings, we wanted to ask the questions: how well do serum plasma bile acid profiles correlate with the duodenal bile acid profiles? And here we saw a very high correlation between those two different sources of bile acids, suggesting that in field conditions that serum bile acids could provide a pretty good readout of what's actually going on, not just in the serum, but within the gut lumen. Okay, so I'll summarize there, and hopefully I've left plenty of time for questions. So that we saw a lot of subclinical cholestasis and bile acid disarray in undernourished children from this cohort that had a very high prevalence of wasting, underweight, and stunting. Interestingly, Robert Bansma, who many of you know from Sick Kids, had done a study of bile acid profiles in children who were admitted to the hospital in Malawi for management of severe acute malnutrition. And he saw a similar increase in total serum bile acids. He also saw specifically an increase in glycolic acid in these children who were around 2 or 3 years of age and were admitted to the hospital with severe acute malnutrition. And so one interesting finding from our study is that we are seeing these in infancy in a community setting. And so perhaps suggesting that these bile acid and liver derangements happen very soon in infancy in the progression to MAM and SAM. We were able to show that glycolic acid and secondary bile acid deficiencies correlated with poor growth, inflammation, and some histologic features of environmental enteropathy. And that serum bile acid profiles provided a reliable proxy of what was going on in the lumen in terms of bile acid dynamics. And what might be some implications of these findings? We think that serum glycolic acid may provide an early indicator of EED and growth failure in infants that reflect subclinical cholestasis, impaired function of the gut microbiota, and enteropathy. I think one important message as well is that we know from a variety of childhood cholestatic conditions that cholestasis impairs the ability of the gut to absorb lipids and fat-soluble vitamins. And so like some of my early work with Dick and Aldo, we showed interesting correlations of vitamin A deficiency with Dr. Blainer at Columbia and enteropathy and lactulose mannitol. And so cholestasis, impairs the ability to form micelles in the gut that allow for proper lipid absorption and fat-soluble vitamin absorption. We also know that the liver is very important for being able to respond to growth signals, and that when there is inflammation, that there is growth hormone resistance. And so, in the absence of healthy liver function, gut-targeted treatments may be less effective in preventing and reversing EED and stunting. And then another interesting potential implication of these is that in other cholestatic disorders in which you're not able to correct the upstream causes of cholestasis for a child, There are a number of studies now showing that providing oral bile acids can improve fat absorption, fat soluble vitamin absorption, and improve growth. And so Robert Bansma is now looking at the role of using ursodiol orally in patients with MAM and SAM to see if you can improve outcomes in that setting. Ursodiol may not be the best choice of an intervention. It's not a really good FXR agonist, and there may be some other promising bile acid approaches that could be explored in a community setting as well to prevent stunting. So with that, I will say thank you very much for your attention. This was a huge amount of work in collaboration with folks here at UVA, Cincinnati Children's, in particular, it's the Central Lab that did the mass spec and the analyses, AKU and their wonderful field team, and being able to pull a study off like this in very resource limited conditions, and lots of inspiration from Dr. Garant's work and Aldo's work in Brazil over the past decades. And of course, thanks to our funders, the Gates Foundation, for the study. And I will stop there, and I'm happy to take any questions from our attendees.
Yeah, great, Sean. Thank you very much. I think we do have a question already from Jonathan Swan. Please, Jonathan.
Great.
Speak.
Hey, Sean, great talk.
Thanks, John.
Yeah, I just wondered if you'd looked at the bile salt hydrolase enzymes or genes in the feces of these kids.
We did not look in the feces, and that's a great comment. We did look at C4 in the serum, but we did not. We have not looked in the feces. There is some nice work from Jeff Predis at Baylor, who has used the regional basic diet, which you're all familiar with, in mice, and he has shown a sort of a fecal bile acid deficiency in those mice, and he's also shown a cholestasis in those mice and some hepatic fat accumulation, and so that's sort of a partial answer to your question, where I do think if we did do fecal metabolomics, that we would be able to verify what we saw in the duodenal aspirates and also the kind of the signature that we're seeing in the serum. I hope that partially kind of
no. I think it's
Okay.
really interesting, yeah, because I don't know if you've seen the work on giving, I think it's chenodeoxygonic acid to. It promotes energy expenditure in brown adipose tissue, and it's shown that it can protect against obesity. And whether that can go the opposite direction and kind of make malnutrition worse, it could be a really interesting area to look at.
I agree. I agree, and I think that the liver and bile acids have been a little bit ignored as sort of target organs in sort of the malnutrition and stunting pathway, and we see lots of, clinically, when we care for malnourished children, we see lots of liver lab abnormalities, whether it's, conjugated bilirubin levels, elevations in transaminases in those children. Sometimes when we're renourishing children after diagnosis of celiac disease, we'll see a bump in their hepatic enzymes while they're recovering. So I think, the liver is sort of this master metabolic regulatory organ. And I think we're going to have to figure this out if we're going to make progress towards this, no child stunted goal by 2030 that we would all like to see.
Thanks.
Okay, so let's see if what still waiting more question, but Sean, let me ask one little thing. When you analyze your design, your design style was controls children with more than -2 z-score for I think it was for high z-score, yeah, and Then you also have the case as malnutrition, less than -2, yeah, equal or less. And then you talk about environmental enteric dysfunction with the deficient on the bioacid secondary metabolites that has been dysfunctioned by the microbiota. Have you looked more specific on those children who had not malnutrition, I mean, but actually more than -2 Z-score, and also explore biomarker of inflammation to see if you also those sustain these deficient of the secondary bioacid metabolites? Maybe
Yeah, that's a
exploration over here.
Yeah, that's a great question. We've not looked at that specifically yet. Najiha Iqbal has been doing, in collaboration with the Gordon Lab at WashU. There was a group of children who were enrolled with very good anthropometry, and they had more frequent stool sampling so that we could get a better sense in the Pakistan context, like what a healthy microbiota looked like in that community of kids who grew well despite kind of harsh environmental conditions. Yeah. And so that characterization continues. We've not been able to sort of replicate this sort of microbiome for age Z-score finding that Jeff had shown in Bangladesh. And there's there's a distinct microbiota there compared to what's been seen in Mirpur and other parts of Bangladesh. And so one other way that this sort of ties back between the gut and the liver is that one of the things that may be irritating the liver or hurting the liver could be bacterial endotoxins from an unhealthy gut barrier.
LPS.
Yeah, LPS. And so, if paracellular permeability is not healthy and that's leaking into the systemic circulation, that LPS may be contributing to some of the liver injury that we're seeing. And so it's a good question that we need to do more to address.
Excellent. Yeah. Well, I think I don't see any more questions. Thank you.
Dr. Aldo,
Pedro,
I do have a question. Yeah.
Yes.
So thank you, Sean. So I do have a question about bacteria. So because there are recent data showing that accumulation of bile acids in intestinal cells could help Shigella to form biofilm in vitro.
Oh, interesting.
And then I wonder if you have any enteropathogens detection data and if you could see any correlations with bile acids. Yeah.
So all great questions. I'm trying to, we did TAC arrays on the children at 2 different time points. And so I would be surprised if we haven't done any of those comparisons, but nothing sort of jumped out significant that was sort of worth reporting in this publication. It is well appreciated that bile acids have some antimicrobial benefits, but the biofilms in Shigella, that's fascinating, and that could really tie nicely into the data that Aldo showed in his talk of those sort of linear growth deficits. And then the second part of let's see, did that address both of your questions, Pedro?
Yeah, sure. And actually that data, the bile acids data inducing biofilm from Shigella is actually from UVA. It's from Hervey Agassi from the microbiology department. I guess, I don't know, that would be interesting to look at.
Absolutely. I'm definitely down the rabbit hole with these. For a long time, we just thought of bile acids were important for digesting lipids, and now we're learning that they're very important signaling molecules and that they have very, very interesting interactions with the gut microbiome. And so Dick and Aldo, know better than I do about sort of interesting correlations between bile acids and C. difficile, for example. And so more to come.
Yeah. Well, thank you very much, Sean. I think we're going to move for the next stop. Now we're going to call Jonathan Swan from the University of Southampton. He's going to approach characterizing the biochemical alteration associated with environment and enteric dysfunction using the approach of metabolomics. John, please get on. Yeah.
Great, thank you, Aldo, and thank you, TMRC, for inviting me to talk today. Okay, I don't think my mouse is working. No, can you see my mouse okay, Aldo?
Very nice, very nice. We see.
Okay.
Yeah, we can see, yeah.
Oh, great. Yeah, thank you. Thank you for inviting me to talk. And yeah, so I'm going to talk about how we use metabolomics really to study EED. And I'm probably showing some data that most of you have seen before, so I'll try and mix this in with newish data as well. Okay. Now, I'm pleased Aldo and Sean, and I think even Alex showed some data on the microbiome. And so I just wanted to kind of get across this idea of the supraorganism or the holobiont, and the idea that we are the collective, the kind of composite, the combined inputs of our own genome and all of those genomes of the bacteria and the various other microorganisms living in and on us. And those interactions between the genome and the microbiome, they have important roles in health and disease, but also in development right the way across the life course. And we've kind of got emerging data suggesting even the mother's maternal microbiome also influences our future development as well. And so today I'm mainly going to focus on this sort of first 1000 days of life region, and using metabolomics then to study this vicious cycle. So just for anyone not familiar, we're doing metabolomics. So when we talk about omics, we're talking about the collective characterization of pools of biological molecules. So the idea of measuring lots of things. And because we're doing metabolomics, we're looking at metabolites. So the idea is we're measuring lots of metabolites in a sample. Now it can be urine or blood or tissue samples. And the idea is by measuring lots of metabolites, we can gain a good overview of all of the metabolic processes occurring within the body. And then we can see what metabolic processes are altered in the presence or absence of EED, or the presence or absence of a particular pathogen. Now, a strength of looking at the metabolites rather than, say, the proteins or the genes is that when we look at metabolites in, let's say, a urine sample, we pick up metabolites that come from the genome, so from the human metabolic processes, but we also see signals that come from the diet and microbes and even pathogens themselves, as well as interactions between these. So we really can gain an overview of both the inherent host metabolism as well as these environmental inputs. And so just kind of talk about these different components of this vicious cycle and how what metabolomics tells us about these different interactions. So again, I think I bored everyone before with this data. So this was a study we did quite a long time ago now. I remember one of my first TMRCs with Dick and Aldo. And that kind of led on to this work, looking at measuring metabolites in the urine of these Brazilian children. And what we did was we identified metabolites that were associated with these different measures of growth. So here, anything in green is higher in children that grow well, and anything in red is higher in the urine of children that were stunted. And one thing we've seen, and we've seen this across multiple cohorts now, is that betaine and dimethylglycine are excreted in the urine in higher amounts by good growing children. And we see this in, like I say, several cohorts. Now, betaine you can get directly from the diet, or it can come from the breakdown of choline, which you get from the diet. And then this betaine gives up a methyl group to drive this homocysteine methionine pathway, which then gets converted into dimethylglycine. Now, because these two metabolites are excreted in higher amounts in good growing children, it gives a strong indication that they've got more betaine available to them, and they're pushing more of that betaine through this pathway. And this pathway is important, and I'll come on to that in a second, but we think the reason for this, the fact that good growing children, or rather stunted children, have lower availability of choline and betaine, we think there's probably a number of factors. One is lower intake from the diet. We think there's some small malabsorption in the small intestine, and I'll show some data for that later. We think that the gut microbiota break down more choline, so they compete with the host. And also some data showing that specific infections can also metabolize this choline and lowering the availability of it for the host. But these are important. As I said, the betaine drives this homocysteine methionine pathway, which is important for generating this metabolite, S-adenosyl methionine. And this S-adenosyl methionine carries these methyl groups with them to different biological metabolic processes. And one key one is DNA methylation. So it gives up this methyl group and it tags genes. And when it tags genes, when it methylates these genes, it turns genes off. Now, this is really important for modifying the developing system, or modifying the genome of the child so that its developing system meets or matches that of the environment. And so this is really important developmental process, and it's dependent on the availability of SAM, which is dependent on the availability of betaine and choline. And we think this is particularly important. It's important for growth, but it's also really important for brain function, this DNA methylation. And in fact, this is some work published last year showing that here we took the forebrain of mice at different developmental phases. So we've got 3 days old, which is the neonatal, 21, which is prepubescent, and then young adulthood. And what you see is in the brain, they have high amounts of betaine, dimethylglycine, and S-adenosyl methionine, all in that neonatal window. So it shows that they need to have a high availability of this in its early time point. And then as we move to progress to the later ages, that really drops off, showing the need for it is much less as they develop to adulthood. So it really does show the importance of betaine, dimethylglycine in this early window for the brain. And as I said, we also think that the microbiome has a role here. So if it seems that the malnourished microbiome has a greater conversion, so this was work published with Dick, in the mouse models, one of a protein deficient diet, that if you protein deprive the mice, their microbiome metabolizes more choline to trimethylamine and dimethylamine. And that trimethylamine then gets absorbed from the gut and converted by the host to TMAO. So the malnourished microbiome does more of this, depriving the host of the choline. And again, we've also measured choline and TMAO in the brain. TMAO seems to be quite an important brain molecule. It seems to be associated with lots of negative things. And we've seen in a model looking at cryptosporidium that actually cryptosporidium seems to also produce TMAO. So it produces this signal, or at least it produces probably trimethylamine, which then gets converted to TMAO by the host. And there's data coming out showing that this TMAO is elevated in Alzheimer's disease. There's also evidence to show that trimethylamine N oxide also promotes brain aging, can increase synaptic damage, as well as inhibiting mTOR signaling. So this malnutrition infection metabolite seems to impact on the brain. And so we think this may underlie some of this cognitive impairment we see, this impaired methylation and also disrupted signaling. And this was a study we did with Christopher Coe's group at the University of Wisconsin, looking at rhesus monkeys. Now here, Chris puts these pregnant mothers on a particular diet, which means some deliveries will be sufficient in iron, so some infants will be born sufficient in iron, and some will be born iron deficient. And then, so these iron sufficient ones stay iron sufficient for the study. And then these iron deficient ones, once they're weaned, they then can either naturally recover. So over a few months, they can naturally recover to become iron sufficient, or they were supplemented with iron to really speed up that process over 1 or 2 months. And then we collected urine, plasma, and feces and looked at the microbiome and the metabolites. Now, just to make the point here, at the time of measuring, all the animals were iron sufficient. It's just some had previously been iron deficient and recovered naturally, or been kind of sped up that recovery. When we looked at the microbiome, there was a modest impact on the microbiome. There were some subtle changes to the bacteria, but not huge amounts to the composition. But when we looked at the metabolites, so here the yellow are the urine and the red are the plasma metabolites. And so here, anything in red is higher in the iron deficient versus the iron sufficient. And here, anything red is higher in the iron deficient compared to the supplemented, anything blue is lower. And what we see, lots of these metabolites come from the microbiome host co-metabolism. So it suggests that there is this lasting effect on gut microbial host co-metabolism. And also there seems to be an impact on the TCA cycle. And we think the reason for this is that these two enzymes here and here, these are all dependent on iron. So even though these animals are iron sufficient, several months later, there are still metabolic perturbations caused by this iron deficiency. So that's even with, in the iron deficient, but iron supplementation can reduce those effects. We can look at those metabolites then in networks. So here, anything with this kind of darker gray background is a metabolite that was altered by iron deficiency, and anything with an orange border around it is one that either its production or its consumption requires iron, and the solid lines are host enzymes, and the dashed lines are microbial enzymes. So what you can see, looking at from the supraorganism point of view, this early life iron deficiency, even after recovery, has still disrupted both the host and the microbial enzyme function. And then we can put the other metabolites then on the background, and you really can see that this is quite a wide-ranging disruption caused by this iron deficiency. So staying with the microbiome then, if we look, so this again was work done with Dick in a mouse model of protein deficiency. We see that early life malnutrition alters the development and maturation of the microbiome. And some recent data on a similar diet, we found that, again, there's these microbial differences caused by protein deficiency, and there seems to be some subtle gender differences as well. Also, in those Brazilian children, we saw that the stunted kids also had higher amounts of metabolites that come from the proteolytic activity of the bacteria. So it changes, stunted kids have a different functionality in their microbiome. And we think that this is both the cause and the effect of ED, not the sole cause of ED, but a kind of contributor. Now, this is work, this was done in Bangladesh, where they found, so normally your microbiome is nicely compartmentalized. You have an immune and physiological factors that keep nice compartmentalization of the microbiome. So you have an oral microbiome, a small intestinal microbiome, and then a colonic microbiome. But what they found is in the stunted kids, there was this loss of control. You had members of the oral microbiota present in the feces, and you had lots of small intestinal bacterial overgrowth as well. And we think one of the contributors to this is to do with malabsorption through, as a consequence of ED. And this was work with Paul Kelly done in Zambia, whereby again, Paul collected duodenal samples, duodenal aspirates from the guts of these people, and measured gut damage with this intestinal fatty acid binding protein. So the idea here is when the gut's damaged, it releases this IFABP into the blood. So higher amounts of IFABP in the blood indicate gut damage. And then we looked at which metabolites, so these are metabolites measured by NMR spectroscopy, and we looked which signals were positively or negatively correlated with this marker of gut damage. So you can see here, anything pointing up is higher in the lumen in those aspirate samples when there's gut damage. And you can see that this is a range of different amino acids and also choline. So what this suggests is that when you get gut damage, either through, with EED, this prevents these amino acids and choline from being absorbed. And so they become more available either for pathogens, but also for the gut microbiota themselves. And we think that this stimulates the, either these amino acids make it down to the colon where they're further broken down, or it's responsible for stimulating that small intestinal bacterial overgrowth. And we've got more data now in children as well, and we see the same thing. We see an increase in lots of amino acids with this gut damage. And what this does, so generally speaking, carbohydrate breakdown by the bacteria produces these short chain fatty acids, which can be beneficial, whereas proteolysis by the bacteria tends to produce compounds which are more harmful, like the phenols and cresols. And this is what we see in the stunted children from Brazil. Now, we're also interested here. So tyrosine is an amino acid found in the diet that gets broken down by the bacteria to 4-hydroxyphenyl acetate, and then gets further broken down to paracresol. And actually, we've got data now showing that Giardia does this conversion as well. And so that suggests that if your bacteria are doing more of this conversion, your gut is exposed, or your body absorbs more paracresol and PHPA. Also, tryptophan gets broken down to indole by these microbes. And so this happens more in the malnourished kids. So then we looked at what would happen if you took these microbial metabolites and dosed them to muscle fibers. And what we observed was that all of these metabolites, which are higher in the stunted kids, they all have a significant inhibition of muscle fiber growth. So then we're kind of playing around, perhaps it's ambitious or a bit far-fetched, but the idea that the microbes are sensing the nutrient availability and regulating the development of the host to match its nutritional environment. One mechanism we think this might act through, so this was again data from Zambia, and so here anything in blue, so this was looking at metabolites associated with mTOR activation. So anything in red is positively associated with mTOR activation. This is in the urine, and anything in blue is negatively correlated. And you can see here that paracresol sulfate that we see in the stunted kids, this is negatively correlated with mTOR activation. So the more paracresol sulfate, the lower mTOR activation. And so this might be a mechanism through which this inhibition of this muscle fiber growth is. Kind of staying on that theme as well, we also see, as I said earlier, we have this HPA, which can also, if I just go back, HPA, this one, inhibited muscle fiber growth. And this is a marker, I've lost it now, HPA there. This is a marker of Giardia. We know Giardia can produce this. We see this in Peru and Tanzania in the MAL-ED studies. So again, can Giardia have an impact on growth through this muscle axis? And then also we have interesting metabolites like this hydroxymethylbutyrate, this HMB. This is nicely associated, it's quite tightly associated with muscle mass, promotes muscle growth. It's an mTOR activator. And we see that the more Shigella have, the lower your HMB is in your urine. And this time, same with EPEC, more EPEC, less HMB. So again, these could be implicated in that muscle axis as well. How am I doing for time? Okay. So then we have this. So we have this idea that a nice healthy gut microbiota promotes gut barrier function, keeps a nice healthy gut, whereas a more harmful microbiome breaks down more protein, produces more toxins, which break this gut barrier and lead to more movement of bacteria and their components like LPS into the circulation, which triggers this inflammatory response. And what this can do is this can activate, this inflammation can activate this tryptophan kynurenine pathway. And so where this comes in, when there's inflammation, this enzyme IDO gets switched on, converting more tryptophan to kynurenine to 3-hydroxyanthranilic acid, which guides naive T cells away from an inflammatory profile towards a more tolerogenic profile. Now, the consequence of this is it diminishes the host's ability to generate Th17, and we think this may contribute then to the persistence of infections in these chronically inflamed children. Also, you get the tryptophan you get from your diet, and this is really important for generating NAD+. So if you've already got low amounts of tryptophan entering the body, either because you're having low amounts in the diet, but also because of that malabsorption effect we saw earlier, then this further inflammation is going to further disrupt your ability to use this tryptophan to make NAD when you need it. And it's been shown quite a few studies now that this plasma KT ratio is higher in the stunted kids, so it kind of confirms this. Now, one thing we saw in the Brazilian children was that those that had a HAZ as at enrollment of -1, if we looked at their growth 6 months into the future, we found that the children that had better catch-up growth had higher amounts of this NMND in their urine. And we believe that this is a marker of the availability of NAD+ and nicotinamide. Now, NAD+ is really important for making NADH. It's really important in TCA cycle making energy. And when this NAD+ is consumed by enzymes like sirtuins, it converts it to nicotinamide, which is then recycled back to NAD+. And so the idea is if you have lots of nicotinamide and NAD+, when you have nicotinamide, the body methylates it and puts it out in the urine. So if you have high amounts of N-methyl nicotinamide in the urine, it suggests that you've got quite a surplus of NAD+ and nicotinamide. And the fact that good growing kids have higher amounts of NMND suggests that this might be a useful marker and this might be a useful pathway to target. And going the other way, it seems that the kids that don't have very good catch-up growth have lower amounts of NMND, suggesting that they've got reductions in this pathway. And then kind of following on from this then, Mark de Boer is leading this illicit study at UVA, looking in children from Tanzania and supplementing them with nicotinamide. Now, unfortunately, the nicotinamide supplementation doesn't seem to have really impacted on growth, but we've got some initial data looking at the metabolites that suggest that the reason for this is that not all kids are in the same metabolic space. It kind of depends upon the season you're born in. And so as you dig into this a bit more and look at in better resolution, you can see that perhaps we're just targeting the interventions wrong rather than nicotinamide not necessarily working. So that leads us on to the last piece then. Are nutritional interventions specific enough? So is one size fits all the wrong approach? And to kind of look at this, we looked at biochemical aging, this concept of biochemical aging in the MAL-ED study. So here we had urine and plasma metabolites in children from Peru, Bangladesh, and Tanzania. And we took a group of children we defined as healthy, as in they had an enrollment LAZ greater than -0.75, and 24-month LAZ is greater than -1.25. And we took these as kind of like the healthy growing kids. And we had metabolic profiles for these children at 3, 6, 9, 15, and 24 months. So we could look at, as you had the ages of these kids, what metabolites changed as they got older. And that's what you see here. Anything in red goes up as the children get older, anything in blue goes down as they get older in the urine and in the blood. And then we were able to pool the healthy sites, and we could get this kind of common signature of healthy biochemical aging. We could then take those metabolites, I forget how many it is now, maybe 20, 21 metabolites, and build an equation where, because we know the age, we can calculate the biochemical age of a child based upon these measurements. So we could take urine from another child, measure these metabolites, feed it into the equation, and get the predicted biochemical age of that child. Now, because measuring this many metabolites in the field might not be that suitable, we then refined it to the 8 most effective or influential metabolites. These were then used to calculate our biochemical age or phenome age score. And so, yeah, so this was used to help create a model where we could calculate the biochemical age. And the idea is then we could take a child, look at their biochemical age, and match it up with their chronological age and see whether they were biochemically immature, biochemically mature, or kind of where they should be. And to make this sort of standardize this to use across populations, we then converted it to a phenome for HZ score, a lot like the microbiome for HZ score. And then when looking at the healthy kids then hovered around 0, as you would expect. But the growth constrained children, so those with poorer growth, you can see that they're statistically lower, almost -2 standard deviations as early as 3 months. They're biochemically immature even at 3 months. So if you're targeting interventions at 6 months, you probably already missed that window. And so this is this idea that we target interventions at chronological age, but not all children at that same chronological age are biochemically the same. And really, we should probably be targeting the interventions at their biochemical age. And it starts to get really interesting when you look at the individual metabolites. So we have these 2 TCA cycle intermediates. Unfortunately, nicotinamide was significant in the Peru and Bangladesh, but not for Tanzania. But this TCA cycle, so showing energy generation, is interesting when you look at length gain measures. And we know that around 35% of your total energy is required for the first 3 months for growth. And then that halves by 6 months, and then it's just 3% at 12 months. And actually, that kind of looks quite similar in the citrate measures and the succinate measures here. So again, if you're trying to target energy metabolism, perhaps you need to do it in this 0 to 6 month window. When we look at the betaine, you can see that really does follow quite nicely the length gain curves. Kind of stressing the importance of betaine during this early period for health and development. And also choline, again, it emphasizes the importance of choline and the links potentially with muscle and choline. I think that's my last slide. So then just to kind of summarize then, this idea that perhaps we need to move towards more individualized interventions, focusing on the biochemical requirements of the individual rather than kind of chronological age. Although I appreciate that's completely easy to say, doing this in practice is a lot harder. So with that, I'll acknowledge all the people that contribute to this work and happy to take any questions.
Great, Jonathan. I think we open for question right now. Let me see here my chat if I can. Before I see any good question here, let me ask you, Jonathan. I really try to think in barrier function and how to move to nutrient to improve microbiota maturity in order to healing or repair the barrier function. How do you approach, have you approached these metabolite or maybe nutrient for the microbiota growth in terms of maturity or health invasion microbiota like Jeff Gordon has been shown before? How do you think? I mean, what are you thinking about this?
Yeah, so we do quite a bit of work on pre and probiotics and postbiotics. So aiming to try and fortify or implant in different bacteria, even starting to look now at bacteriophages, so the viruses to try and modulate the bacteria through those. I've lost my train of thought where I was going. Yeah, so there are mechanisms to modify the bacteria. And I think we're starting to learn more, and even kind of what Sean was showing, we're starting to understand more about the small intestinal microbiome and why they're important and how they're important. I'm really interested to study that whole microbiome infection competition effect. Yeah, we're doing some work with Paul Kelly actually on the microbiome of those duodenal samples. And yeah, it's trying to make sense of it all, really. You have so much data and trying to pick out one particular bacteria that's important. There's also the question, and maybe Sean might be probably better place to answer this, perhaps they have the microbiome that they need. You could argue you have an aberrant microbiome that has harmful effects, or you could argue that there are microbiome that are suited to that gut. I don't know. We have worked, I haven't seen much on this, but when I was at Reading, we had a lot of these gut simulators. And again, I don't know if Sean's got these set up in his lab, but this idea where you have batch cultures of fecal samples from individuals, which you can then dose and you can see how they respond. We've done it with bile acids and it works really nicely. But yeah, I think that would be a really nice way to go. And again, that could be something set up in Fortaleza as well.
I think Sean has some questions, but let me add one more thing. I have seen acetate and butyrate as a good metabolite from the microbiota that has been shown to improve, barrier function, and also having some immune system stimulation that improves some infection. That I have seen 3 papers right now showing that the acetate is a very good metabolite from the microbiome who helps to control the syncytial respiratory infection. I was very impressed about this. And I think, Jonathan, something that could improve those microorganisms to synthesize the short fatty acid, I think would be a nice approach to repair or to improve intestinal barrier function. But let me see if I'm thinking correct.
Yes. Well, that's where prebiotics often have their effect. They're broken down. They stimulate sort of beneficial bacteria to break them down to short chain fatty acids. And most of the data you see is sort of a beneficial effect of the short chain fatty acids. There's the odd study kind of showing negative effects, but generally speaking, they can be good for inhibiting pathogen growth as well by acidifying the gut, improving gut function, improving immune function. I think there was one study showing that the short chain fatty acids actually improved muscle growth as well in athletes.
Excellent. So, Sean, have any questions?
Yeah, just a tag on comment, and then I'll ask John a question. That you have that sort of intact fermentation capacity and the ability to make short chain fatty acids in the colon. And specifically, there's lots of interesting data about butyrate being able to induce differentiation of T regulatory cells in the colon, and there's been a lot of interest in their ability to prevent colon inflammation. So, John, for a long time, people have used folate supplementation and antibiotics as a sort of a tropical sprue therapy, and folate's also sort of interesting as a methyl donor nutrient that could probably affect sort of epigenetic and gene expression in addition to providing nutrients. And I'm wondering, has anyone looked at choline supplementation, or the groups? I think in a group in Colombia and some others have seen a really nice effect of giving an egg a day. And so eggs are packed with choline. And so have you ever got a hold of any metabolomic data from egg studies, or do you think a choline study would make sense?
I've forgotten. I think is it Chris, somebody at in California, Christine Stewart. Is it Christine Stewart? I think they did a study in Ecuador which showed a beneficial effect of eggs. And at one stage we were going to look at the metabolite profiles of those. It never really took off, because I know they repeated it. I think they might have repeated it in Malawi, and they didn't see an effect.
Oh, interesting.
But yeah. That could be a gut. If you've already got malabsorption, perhaps it doesn't matter how much choline you put down there. might be one mechanism. And I think Andrew Prentice is just starting a study looking at seasonality and epigenetics and meat-pole donors. So again, that might be some interesting data might emerge from that.
Very interesting. Great talk. Thank you.
Yeah. Well, thank you, Jonathan. I think we're going to move for the last talk for this section. I think we have a chance for delay a little bit the last section 3 on Leishmaniasis disease, because Dr. Selma Jerome jumped for the tomorrow section. I think we have a flexibility for Dick to, because our time here we have actually 20 minutes ahead of time. Ahead now, I'm behind. So Dick, please, I don't need to introduce you. You are the leader who actually grow everybody here. And please go ahead and talk about your modeling enteropathy or diarrhea with the top bacteria and protozoa pathogen. Update on differential determinants of outcomes. Please, yeah, you want.
Okay, Aldo, do I have my full screen?
Full screen? No, not yet. Full screen now. Yes, you do.
Okay. Well, thank you. I actually was trying to ask, I'll use the best little piece of my time to ask John to come back and tell us whether the SAM story that he was sharing might explain specific effects in the brain that may be independent of growth. So if you're still around, John, I'd love to hear that. And my other question that I was trying to get to was to ask short-chain fatty acids for the colon is sort of like alanyl glutamine for the upper bowel, if there's any promise for that combination, perhaps. Anyway, thank you. I'll get going because this really is a very special opportunity. Aldo, thank you for inviting me, and this is obviously something that that you and Ronaldo and Alex are certainly a part of. John is a major part, as you know well, and Sean over many years. Others here as well that I'm going to come back to, but Pascal in South Africa, but David Bolick, Les Rogowski, and James Platsville, Eric House, Jim Natero, and others. In fact, I think it's Dr. Swan's great review of animal models that sort of has a lot of what I'm going to talk about with the references. But let me first just say what a tremendous honor this is to be celebrating, Aldo. I think this is the 25th year, the 24th TMRC meeting. Only last year it got missed. And actually it's more than the 45th year of the Sierra Natal Bahia UVA collaborations. And looking back on this, I hadn't realized that it was in 1977. Actually, it was my birthday that Ed Hook and I visited for 3 days for the first time, Fortaleza, and Dr. Galba Araujo and Auxiliadora de Souza. And in those 3 days, met Nogueira Paes, Manassés. I don't think we met you that day, but quite yet, but that was the prodrome. And soon followed Anastasio, who will be in the next session, you and Noelia, of course, Reinaldo, and so many other wonderful people. So this thing popped up in the middle of my screen. How do I get it down there? There, now it's out. Okay, so that I can see Shirley and Selma and tremendous people that have most recently been Pedro and currently Daisy, just amazing people. And your field team, Aldo, is like no other in the world. We've got a few interesting characters. I mean, here is Barry Marshall in your lab, Fred Murad in our lab with many people, Honey and others. Actually, this was Nancy with Robin Warren 3 years ago when we were in Belfast. Some other interesting people, but most importantly are, again, I got to get my thing out of the way, but I hope on your screen you can see that Selma and Noelia and Aldo, I'm pretty sure that that's Anastasio over here, people like Leah Barrett, clearly David Bullock, Charlotte Martin, our dear Lorna Borey, and John Swan with us in London, and of course Dick Pearson and Surly and so many others. But what I want to do is to, by way of thanking everybody in this group, share 4 take-home points, and 3 are clinical. And everybody has set this talk up for me to just move quickly through it, because we've talked about not only the mortality, but the morbidity with the decline in height for age Z over the first 2 years of life. I think we need to name these new diseases, because until we name them, we won't measure them and pay attention to them. The cognitive hit that we'll talk more about, the metabolic syndrome that we'll talk more about, and then especially, of course, the laboratory models that help us dissect the mechanisms and develop biomarkers and interventions. And so this is a familiar slide to most people in this group, I'm sure. And Jamili introduced this morning the comparison of the still awful, I think it's down to 500,000 now, Aldo, but it's still over 1,000 children a day who die. But compare that with 150 million, 300 times more, who are moderate to severely stunted. And that actually constitutes nearly 1 in 3 children in impoverished areas, actually 1 in 5 children around the world. And here is just that same story showed with the deaths from diarrhea worldwide since 1990, and this fantastic drop in the mortality in under 5-year-old children. But look at what's left here. Guess who worldwide is dying the most from diarrhea of all things? Is 70 plus. If you add the 50 pluses, you get even more. And so it's changing over time. And that becomes amazingly relevant, because like Bucky Greeno showed over 30 years ago now, there are lots of enteropathies and diarrheal illnesses in our nursing homes and long-term care facilities. And so when we think about the effects on not growth, but on cognition and on metabolic consequences, quality of life comes in a fairly major way, because not only when Laurie Archbold looked at just 46 convenience samples, patients in a nursing home here in Charlottesville, she found that 11% actually had diarrhea, but another 50% had totally unsuspected liquid or unformed stools. The people who changed the bed sheets knew, but nobody else. Jim Hughes found the same thing up on our hospital wards many years ago. In addition, 57% had totally unsuspected enteritis with elevated fecal lactoferrin. And some had C. diff, but many more than just that. So it's a huge problem when we talk about inflammation that I've kept asking about through the day, not only in its associations with linear growth and the pathways involved, but perhaps also the cognitive effects and perhaps sometimes independently with cognition and metabolic consequences. But Aldo, it was your studies in Ippredi that showed so elegantly that with Mark DeBoer helping us put this all together, that if the children had fever, cough, or diarrhea recently, or a fecal myeloperoxidase elevation, each of these independently of the others was highly significantly associated with systemic inflammatory markers, the HSCRP. And that had the effect that that Sean and Jonathan and others have already mentioned, of impairing the hepatic response to make IGF-1 and IGF-BP3 when trying to be stimulated by growth hormone. However, it removed the negative feedback, and that's why there's actually a positive association with growth hormone, trying to get the liver, which isn't responding, to make the growth factors. And so it's been Jims and Mally D that so thoroughly have been studying these causes of diarrhea, but Mally D looked, of course, beyond just the acute illness and found that the pathogens that are associated with impaired growth, cognition, and later effects, even metabolism, tended to be different from those that were just associated with acute diarrhea. The viruses almost all drop out, with one or two exceptions, and it's the protozoa and bacteria that are most important. In fact, it's that whole pathogen burden, as well as fecal myeloperoxidase now, and Shigella specifically, that is significantly associated across all the MALAD sites, even without any diarrhea, and with impaired cognition. And those are new unpublished data that are in have been submitted. I think probably are tentatively accepted. Anyway, we can model in the mice diarrhea, growth, maybe even, thanks to some of Daisy and Reinaldo's work, some of the cognitive and even metabolic effects. And we can do that with all 7 major pathogens that are associated with these longer term consequences. And so when we add in diarrhea and LM inflammation, I'll mention briefly the data that associate in children with subsequent metabolic syndrome. So we were talking about those asymptomatic. I don't like the term. I prefer the term silent, because they're not asymptomatic. It's just we haven't paid attention to the long-term consequences. But if we look at the pathogens that are associated with fecal inflammation, myeloperoxidase, intestinal inflammation, in over 20,000 stools, I think this was from more than 1,400 of the children that had all the data. And clearly, number 1 is Shigella, followed by Campi. And there's our friend E. coli, just for Alex. I'll come back to EPEC. Norovirus slips in as number 5, and ETEC is still all highly significantly off of the into the highly significant space. Giardia is on the opposite side, associated with growth failure, but negatively associated with the inflammatory response. So there's a different mechanism likely there than the inflammatory one. What else that means, we really need to keep understanding. But in addition to diarrhea, we talk then about the effect of enteric infections, either directly on growth stunting or has drop, or on the cognitive impairment, cognitive hit, either again directly or indirectly through stunting, some indirect, some independent of stunting as well, we think. Similarly with metabolic syndrome, it can either have a direct effect or effect through the pathways of stunting or obesity. And each of these has references noted to note those. Well, I've showed this many times because it's so important to appreciate Aldo's long-term follow-up of these precious children in Gonzalez Diaz, who after we had followed them from birth for the first 2 years, we found that 4 to 7 years later, they had cognitive impairment that was specific to the semantic fluency. And it could be as much as 10 IQ points. This is with the average diarrhea burden that these children had suffered. And of course, it was Reinaldo who helped us sort out that this E4 gene allele is actually protective, the opposite of what we anticipated, because it's the biggest risk for Alzheimer's disease. And this positive effect in children, protective effect, may explain, like malaria explains sickle cell disease in the population, it may help explain E4 in Alzheimer's and Alzheimer's disease. In fact, that E4 allele can be targeted transgenically inserted into mice, and Orleancio found that indeed those mice had in better growth and better villus to crypt architecture, as well as shedding less cryptosporidium. Again, these are familiar to most people, but everybody in this group who is over 2 years old, I have bad news. You are on the downside of your synaptic density. We Homo sapiens form virtually all of our synapses in our first 2 years of life. Yes, we gain a few more, but we trim even more for the rest of our lives. And so that's the critical period, though, when synaptic tracts are laid down and when we, Homo sapiens, children, are most vulnerable. And beautiful data showed this in the murine model with malnutrition impairing hippocampal synaptogenesis. That could be reversed. This is hence one of my questions about glutamine with Jonathan a couple minutes ago. So that was also shown by Aldo in the children. This was a huge study over several years, actually, where Aldo was able to find that indeed the children who had been on a zinc, vitamin A, and glutamine arm of this trial indeed had significantly improved verbal learning scores. However, all of this effect for all children was accounted for almost entirely by the little girls. It was gender specific. So I know Sean is working on a gender paper in some of the models, and this has yet to be adequately understood, but is obviously important. Here's the one slide that shows the data from humans, from children with metabolic syndrome following either early diarrhea. And so Mark DeBoer went down to Guatemala, where the old INCAP studies had been done 30, 40 years ago, and found that indeed early childhood diarrhea had been significantly associated with metabolic syndrome in those individuals, now 30, 40 years later. Furthermore, Margaret Kosek with Gwyneth Lee was able to show that CRP in the Peruvian children was associated with increased blood pressure and in later life as well. So this is probably an important long-term problem along with the cognition and growth failure. So now to finish with the mirror, desculpe, eh? No problema? Hello? Okay.
It was a microphone on for somewhere.
Sorry, okay. Interrupt if you need to, thanks. So I will spend the rest of my time really just summarizing by pathogen some of the effects in our murine models, where clearly there are three major determinants among others. And I will focus, the ones that we've studied are differences with different pathogens, differences with different microbiome effects and antibiotics, and differences with protein or zinc in the diet. Jonathan, we haven't studied iron nor an awful lot else, but that needs to be studied. Suffice to say that the responses to all of these differences is different with every single pathogen. The protozoa aren't worse with antibiotics destroying the microbiome. However, one needs to give antibiotics to get a robust model of all of the bacterial infections. Crypto, it made no difference. Actually, Giardia grew better when you killed off some of the microbiome. That's a separate story. In contrast, protein, but not zinc deficiency, was tremendously important in determining the growth and some of the Hasdrop effects of the parasites as well as the bacteria. In contrast, zinc deficiency for some, but not all, of the bacterial pathogens really made them much worse. And we will talk about Shigella and Campi, but it's also true of Adragordiv E. coli, as has been mentioned earlier today. So starting with Cryptosporidium, and I'll just give these examples, by the way, for Crypto, ETEC, Shigella Campi, and EPEC. So starting with the Crypto story and work that was done by Bruno Coutinho and others, it's clear that Cryptosporidium is both a cause and an effect of malnutrition, or is worsened by. So nourished, uninfected, infection alone has this effect. It's even worse with malnourished mice. This is clearest with the histopathology. Malnutrition or infection have effects, but malnutrition and infection together is devastating. And one of the reasons is that the malnourished mice, given the identical Cryptosporidium challenge at the same time, if they were malnourished, they shed not 1 or 2, but up to 5 or 6 logs more Cryptosporidium. Well, we've learned about another new host defense that no one pays attention to. It's called epithelial cell turnover. That's tremendously important for Cryptosporidium. Cryptosporidium normally will drive both apoptosis and proliferation to get rid of this mucosal infection. However, malnourished mice have both apoptosis and proliferation severely impaired. And so that was a really important new host defense we need to pay more attention to. And as Jonathan has far more elegantly mentioned already, we can, thanks to his laboratory and Jordy and Greg Medlock, we can say that in the model that it is trimethylamine oxide that is increased. And by looking also at the microbiome here, we can say that it is not microbial, but it's host derived. And that's actually surprising that so few bacterial composition effects were seen with cryptosporidial infection. But why TMAO? Jonathan has already mentioned that that is the single greatest predictor of cardiovascular disease and death from heart attacks or strokes. And so we eat I thought about this, Sean, with your 1 egg story, but it's the red meat and egg yolk phosphatidylcholine that the gut flora convert to choline. And then, as Jonathan has so eloquently shown, also down the road to trimethylamine. and through hepatic flavin monooxygenases to trimethylamine oxide, and malnutrition, the ApoE story and cryptosporidium can influence that pathway. And this is at least a plausible pathway for some of the longer term metabolic consequences from early life enteropathy. So let me turn to a couple of the bacterial pathogens before closing. And ETEC is fascinating because our most robust model is with perfectly well-nourished on house chow, mice on house chow. And we get diarrhea, and the biggest acute effects of ETEC and zinc deficiency and protein deficiency don't make it much worse. In fact, they shed less than the house chow mice. However, the zinc deficiency was shedding less in these mice if they were LT producers, despite the fact that they had CFA and ST in the ETEC. However, if we knocked out LT, we found that ST only has an opposite effect on shedding and on severity. And the fecal biomarkers are worse with either protein or zinc deficiency. And so let me return to that slide. Remember that Shigella is the worst single culprit for inflammation in the gut. And so a model for shigellosis is important. And we find some of the same findings that normal nutritional status gives us the most robust initial response. However, now zinc deficiency is what really prolongs this infection, prolongs this worsens the shedding, and some of the biomarkers by far. That enables us, though, since we can look at Shigella or ETEC with basically the same model, we can look at effects of bivalent vaccines like this one with Eileen Berry, in which she had a Shigella flexneri derivative that is avirulent with ETEC virulence traits, CFA1 and LTB. And there, Shigella infection not only was protected by the vaccine, but even by the vector because it had the same Shigella traits to immunize. In ETEC, it was only the vaccine, of course, that was effective, but the same vaccine could protect against either infection given the same model. Similarly, we've looked at other combination vaccines, and this Shigella truncated mutant with DMLT also reduced Shigella-induced weight loss, shedding, and diarrhea, and some of the enteropathy scores. So let me turn then to Campylobacter, which is, again, totally different. Without zinc deficiency, we really don't get the ability to colonize beyond just the first few days. But with zinc deficiency, Campylobacter goes crazy. It's a worse growth outcome, and it causes overt bloody diarrhea, clearly more biomarker expression, and striking histopathology. That gave us a model to test Campylobacter vaccines. And so there's been a fantastic collaboration with Christine Zemansky and their Harold Nothaf's Campylobacter in glycan hepatosaccharide vaccine. And sure enough, it was partially protective and gave good antibody responses when expressed in an E. coli. Interesting, this was one of those avian strains that I heard Alex mention a few hours ago. So finishing up then with the EPEC story, it is Ellen Ledwaba, who is now back home in South Africa, but who worked a lot with Daisy and with Pedro and David Bolick, showing a really impressive model with long-term effects that were both typical EPEC and even BFP knockouts that are perhaps analogous in part to atypical EPEC. It's not atypical EPEC, it's a genetic BFP minus strain, but it raises interesting questions because the EPEC strain is dramatic in its destructive effect. And that destruction is every bit as bad with or without that bundle forming pilus knocked out. And so Ellen now has that in press, and thanks to that Trabulsi Symposium, Aldo, I think that you were present for. And the other thing that Ellen has found that is very interesting in light of some of the earlier discussions today, and that is that EPEC, when co-infected with intra-aggregative E. coli, shows an inhibition of all of these marker expressions of EPEC, marker virulence. However, when it's combined with ETEC instead, there's an opposite effect. And all of those biomarker expressions, whether it's EAE or any of these others, highly significant for ESPB and EAE, is increased, but there's a trend that goes similarly across all of these EPEC virulence traits. So in closing, let me just show again the familiar cycle that we all struggle with, because either directly or indirectly, there can be effects with or without diarrhea on cognition, on metabolism, and of course, if we count the poverty of those effects, we find a vicious cycle of poverty. And so I'll finish with this slide, Aldo, if it's okay, to simply say that HESDRUP, CogHit, and METSIN remain important. I'm fascinated that some traits have become negative in their effect over time, like sickle cell and ApoE, that were once advantageous. And so evolution itself is changing over time. I've written a little sequel to that first book, but what's really important is these kids that really have us all at this meeting and trying to solve these problems. Thank you. I don't think I went too much over my 35 minutes. Sorry. Thanks.
Oh, that's great, Dick. I think you package so many models that has so many potential to be explored in terms of virulence traits for these pathogens. It is amazing. I mean, 10 years ago or 15 years ago, we didn't have any of this model possibilities. So congratulations for your huge work on this last decade developing this model based on the clinical data from the malaria. I think that's the real important thing. So I saw Sean raising the hand for a question. Sean, do you want to start?
Those were clapping hands, so no question.
Happy hand. Okay. So,
thank you, Sean.
I was Dick. Let me ask you quickly because we're going to for the next section soon. The Campylobacter you mentioned that was a strain from avian. Is that my understanding correct?
Yes, it was, Aldo, but it's supposedly non-pathogenic and has cleared all the hurdles to be used as a potential vaccine. I just found that origin kind of intriguing. I think Alex may have been the person that mentioned an avian pathogen, and I had to study that a couple of years ago a little bit more deeply. But yes, it does have that original history. that I remember fewer of the details of now than I had at the time. But it is a cleared vector for safe delivery to people,
And
and tried.
one more comment instead of question, because I saw this morning Samilin developed the new diet. The new diet based on malad was low in zinc, and I think also in protein, as she mentioned early morning today. So I think it's consistent with your showing here. And I also think about children having those deficiencies who actually could be something that could be targeted well and easily in terms of prevention and support for those diseases, especially the silent one. I mean, we have a lot to go in terms of treatment and prevention, but I think this is the group and others group that need to be think about these type of intervention. Any further questions? Because I see Jonathan say obrigado, parabéns. Jonathan is speaking already Portuguese. That's good. I mean, it's getting there. So I don't see any
Dr.
further
Aldo,
questions. So
Dr. Aldo, I do have a question.
yes, Pedro.
It's Pedro.
Go ahead.
Yeah, to Dr. Guerin. So Dr. Guerin, so taking in consideration Dr. Aldo's talk and also Jonathan Swan's talk, where they highlight the importance of microbiome, I wonder, I know we've been working on those pathogens mouse models for a long time, but I wonder, what is your thought? What are your thoughts on mouse models that target the microbiome, that we could modulate microbiome to model EED? If you think they could be more useful or less useful, and why?
Great question, Pedro. As you know, and we've probably talked about over the years, obviously mice are not children or people. And it's very important to always remember that. However, what mice can do is tell us what specific organisms might do in vivo, because a test tube is even less like a person. I have been amazed at how similar some of the manifestations, even clinical manifestations in the murine models, match up with what we do see in the children. But I think we've always have to be very cautious about that. But when Jonathan, for example, finds similar metabolic pathways in the children and in the mouse models, and if we then can manipulate those in the mice and then test them, go back to children. I mean, I always try to say we have to move from the field, needs to direct at least some of our important research in the laboratory. But then we need to take that back to the field to test it back and forth. And so I think your point is a very important one. We have done quite a lot, much of which you know about, some you don't, about the different Vedanta microbiome compositions consortium, and a lot of the C. diff work has shown that certain microorganisms can be very interesting. Some of the work that I was alluding to earlier in addressing John Swan's comment is from the C. diff work, and some very interesting work that Jay Shen is doing here with Surly on the aging mice, and what is it that's different about their microbiome that makes them more susceptible, because again, it's just like people. The aging mice are more susceptible, and the young mouse fecal transplant helps make it better. And so zeroing in on what it is that's helping is something that the mice can help us do. And then we need to take it back to the patient, though, before we can say that it's clear. Thank you. And thank Sean and John and everyone for this, and Aldo, this is all your work. Thanks.
So thank you very much. I mean, I enjoy this section has been quite exciting, and I'm very happy with all of you, and also your talks, brilliant talks, inspiring new ideas, and what we have to do more and more all the time. I mean,
John has his hand raised, Aldo.
yeah. I think. Oh, John, yeah, go ahead.
Yeah, I just wanted to make a quick comment. I think Dick hits it on the nose about the importance of eventually, adding microbiome more and more into these models. And we were really excited to be able to recruit Carrie Cowarden here from Jeff Gordon's lab 2 years ago to UVA. And so Carrie has established a really beautiful model where she takes microbiota from stunted children, puts it into germ-free female mice, and then those mice can get pregnant, and then they pass on the stunted microbiota, at birth to their pups. And so she sees very different immune development in those pups. And so I think one thing that we've missed out on a lot of our models is it's easier to work with weanling mice or, mice that have been weaned, but like some of these like first 1,000 days of life models where we actually, we start with the maternal generation, I think we're going to learn some good insights from those things. So thanks for this session, Aldo, and look forward to a great rest of the TMRC.
Well, let me ask you before I close this section to turn on your video, because we need a picture. It's like a picture recording who is present. Sometimes people ask us for the certificate, so we have to know if they were in or not. So, and plus is a memory report. Yeah, please turn on your video quickly because we need to move for the next section.
For the picture.
I thought you'd written a song, Sean.
I'm going to play a little Djavan, maybe some Caetano Veloso. We'll see.
Not a bad idea, yeah. Deu certo? Okay, the second page is going to be take a picture. It's 40 people almost, yeah. So it's a good section. Thank you very much, all of you. I mean, speakers and also the audience to being here. So we're going to move for the next section. Thank you, guys. I appreciate you being here. So let me see the next section. I think I have section 3. I mean, it's leishmaniasis, a common endemic disease, epidemiology, clinical immunology in general. We lucky Professor Selma Jerônimo move on to for this tomorrow section. She's in the airport taking the plane, so she could not make the talk for this afternoon, but she's going to be the last one tomorrow morning. So, but then we have Professor Anastácio Queiroz, Dr. Adam Lima, and Dr. Rubens Lima Montenegro. I'm going to move first to Professor Anastácio, then to Dr. Rubens Montenegro, and the last one is going to be Adam Lima. So let's going to start to present Professor Anastácio Queiroz Souza. Was one of the first postdoc fellow at the University of Virginia. She's a well-known infectious disease here in Fortaleza and has many activities, not only in research, but also public health. Professor Anastácio, welcome.
Thank you very much, Aldo. And first of all, I'd like to give my best regard to Dr. Guerrant, who I owe very much. Dr. Garant, I met him, I think, 1979 or 1980s. And since then, he has helped me over this year in many moments. His friendship and his kindness and his help will be always in my heart. And I'm very happy to see him talking with the same quality, with the same details, with the same knowledge. Thank you very much, Dr. Garant. God bless you today and always. My regard to everyone present. I think there are many important representatives of the institution, and I'm glad to be here, and let's begin. I'm not sure if I...
Yeah.
Não compartilhou ainda não, né?
No, not yet, Anastasia. Yeah.
Deixa eu ver aqui, peraí. Compartilhar a tela, compartilhar, abrir preferência, o que está acontecendo? Não estou...
Vai compartilhar. When you click the share screen below, go to the below icons.
Não, aqui,
Pega o seu mouse e...
Compartilhar, pronto, cliquei em compartilhar.
Sharing screen, né? Compartilhar.
Compartilhar. Aí eu clico em compartilhar.
E aí aparece
abrir preferência.
Selecione a tela que você quer compartilhar.
Pronto.
Isso. Clique nela.
Nada ainda, né?
Não, ainda não. Mas...
Eu estou com o PowerPoint.
Você abra primeiro o seu PowerPoint, isso.
Eu abro só parcialmente, né?
Precisa botar full screen ainda não. A parcial, vá embaixo da sua tela, deve ter aí Share Screen.
O meu tem compartilhar, Share Screen. Ok. Cliquei, aí tem a palavra compartilhar.
Aparece várias opções.
Aparece desktop, aparece quadro branco. Eu não tinha ainda, é um computador novo, eu não tinha ainda usado, mas é um Macintosh. Quando eu clico em compartilhar, aqui está aqui o meu.
Você pode ver alguma coisa aí, porque você usa mesmo.
Aí aparece abrir preferência do sistema. Eu permitir que o Zoom compartilhe sua tela. Abrir preferência do sistema. Eu clico em abrir preferência do sistema, aí vem uma série de... Eu não sei se eu tenho que olhar o PowerPoint nesse...
Se tem alguma tela do PowerPoint aí, você clica nela.
Não, mas não tem PowerPoint.
Parece.
Pode pegar o PowerPoint aqui, vai para compartilhar e selecionar a tela.
Você abriu já o PowerPoint?
O PowerPoint está aberto, não em full screen.
Certo, tá bom. Então... Deve ter aí, nesse share screen, quando você abre, deve ter...
Fazer de novo.
Várias opções de
quando eu abro, eu compartilhar, aí eu clico em... aqui o PowerPoint, eu clico em compartilhar. Aí tem abrir preferências do sistema.
Acho que não, né?
Eu boto cancelar.
Não. Se cancele, vê se tem outra opção de desktop, né?
Mas aparece de novo, ele diz, permitir que o Zoom compartilhe sua tela, abrir preferência do sistema...
Permitir, sim, permitir.
Mas ele não tem permitir, ele permitir abrir, ele manda abrir. Aí aparece uma série de ícones à esquerda, mas não aparece PowerPoint, não aparece...
Não aparece
Coloca o desktop.
Então, na só aparece o desktop.
Clique no desktop.
Só um minutinho. Vamos de novo, né? Que coisa, rapaz. Aparece desktop. Eu clico no desktop.
Sim.
Aí o senhor já pode abrir o seu... Quando ele abrir, o senhor já pode abrir o seu PowerPoint.
Porque ele está usando o Mac, aí o professor está dizendo aí no chat, que o Mac está solicitando atualização e preferência do sistema. Tem que ticar PPT, aí ele está atualizado.
Isso, talvez eu possa ajudar. Vocês me escutam, né?
Não, Zé.
Professor Anastácio, provavelmente é a primeira vez que você conecta essa versão do Zoom no seu Mac. E aí, em preferências do sistema, vai ter uma lista de programas e o PowerPoint vai estar lá, só que ele não está assinalado. Tem que clicar lá no quadradinho para o PowerPoint ou para o Zoom, nesse caso.
Ó, eu botei aqui, pronto, vamos ver. Aí eu boto compartilhar, aí eu boto abrir preferência de sistema. Aparece uma série de coisas.
Vai lá em preferência de sistemas.
Não, eu já tô
lá
aqui
na maçãzinha à esquerda, quando você clica, aparece preferência de sistema, né?
Eu não cliquei em maçã, eu cliquei em compartilhar a tela, aí aparece compartilhar e aparece desktop em azul. Certo? Aparece o desktop num quadro em azul. Aí eu clico nele, certo? Aí boto compartilhar, já estou com o PowerPoint aqui. Aí tem permitir que o Zoom compartilhe sua tela. Abrir preferência de sistema.
Isso. Abriu preferência de sistema já?
Até aqui está aberto. Aí aparece serviço de localização, contato, calendário.
Isso, vai ter o Zoom aí, vai ter o ícone do Zoom. Em algum momento vai aparecer o Zoom. Desce a lista.
Certo.
Que deve ser um dos últimos.
Deve ser um dos últimos.
Aqui aparece análise de melhoria, publicidade, automação, disponibilidade, Bluetooth, HomeKit, mídia, Apple Music.
Vai até o último, porque é o Zoom.
Eu estou no último, análise de melhoria, aqui não tem. Certo? Vamos ver aqui. Aparece um Zoom aqui. Tá ruim mesmo, sabe assim, não tem. Porque eu venho de cima, ó. Contato, calendário, lembretes, muito estranho.
Fecha a preferência de sistemas, clica lá na maçãzinha novamente, eu vou tentar te seguir aqui.
Na maçãzinha do computador?
Lá em cima, canto superior esquerdo. Certo. Aí vai ter preferência de sistema novamente. Aí você entra nela.
Pronto, aqui tem, pronto.
Aí vai ter segurança e privacidade. Security and Privacy.
Estou vendo, pronto. Okay.
Aí vai ter uma lista, né? Location Services, tem uma lista aí, não tem?
Tem serviço de localização.
Location. Isso, aí tem contato, calendário. Lá embaixo tem uma acessibilidade, viu?
Lá embaixo tem. Embaixo tem análise de melhoria, publicidade da Apple, disponibilidade do usuário, Bluetooth, subindo, né? Não tem acessibilidade. Tem aqui, sobre serviço de localização e privacidade, ao lado de uma barra.
Serviço de localização tem uma setinha azul, né? Aí depois tem contatos, correto? Calendário.
Não.
Você está seguindo essa lista?
Não, tem serviço de localização.
Então,
calendário.
aí vai ter uma bolinha azul com um bonequinho.
Tem acessibilidade, é isso?
Acho que você acabou de compartilhar.
Fui eu.
Ah, tá.
Para poder agilizar, para ele poder ver o que eu estou fazendo.
Isso, exatamente. É isso aí o que o Alexandre está mostrando.
Eu não vi o que ele fez.
Lá em acessibilidade...
Eu vou fechar e fazer de novo, professor.
Acessibilidade? Pois não. Deixa eu olhar.
Eu fui na maçã, certo? Cliquei em preferência do sistema. E na hora do preferência do sistema, porque isso aqui está me atrapalhando.
Esse é meu?
Não, isso sou eu.
Certo, ok.
Tá? E aí, quando eu cliquei na preferência do sistema, certo? Eu já estou na preferência do sistema. E aí eu vim em privacidade.
Certo.
E aí aqui...
Acessibilidade.
Encontrou?
Não,
é isso mesmo, Rubens?
Acessibilidade. Aí depois de acessibilidade.
Aí, quando clicar em acessibilidade, clica lá, Alexandre, por favor. Aí vai aparecer, tá vendo o Zoom ali, ó? O Zoom está com um tiquezinho. Aí tem que autorizar ali, naquele momento.
Ok, lá
mas para isso ele precisa clicar, nesse cadeadozinho daqui. Colocar a sua senha.
Isso eu não tenho senha.
Tem que ter. Do seu computador. É a senha que abre o computador.
Sim, tá certo. Aí dá certo. Vamos lá, ver se dá certo. Compartilhar, porque agora, certo? Você não pode... É porque você ainda está... Você tem que fechar o seu.
Eu vou fechar agora.
Certo. Então, pronto. Compartilhar a tela. Compartilhar. Eu vou aqui no... preferência de sistema. Fazer de novo, né? Tá. Interessante, não tá. Agora na preferência de sistema. O Zoom deseja gravar, abrir preferência de sistema. Que coisa. Acessibilidade. Zoom. Aí eu clico no Zoom, privacidade. É privacidade e Zoom?
Privacidade. Tem que estar apto para clicar no ícone do Zoom, depois da acessibilidade. Se o cadeado não estiver aberto, ele não vai permitir.
Outra possibilidade, Anastácio, é você mandar pelo e-mail para mim, que eu abro aqui.
É muito pesado,
É
muito
muito
pesado.
pesado.
Posso tentar, mas não dá, não. Vou tentar novamente aqui. Está muito ruim. Mais um minutinho, pronto, senão a gente...
Tá.
Compartilhou, não?
Ainda não.
Eu fiz todos os passos que o colega fez. Ele não pede nada de... Ele não pede o CAD. Agora sim, peraí, ver se vai dar certo. Eu clico no Zoom, aí eu clico no PowerPoint? É isso?
Peraí. Não. Anastácio, deixa eu fazer uma sugestão. Eu vou colocar você com alguém para gatilhar os seus slides.
Você
Em
continua aí.
seguida, e agora eu vou pedir o Rubens para entrar, porque senão a gente vai atrasando
Ok,
e
tá
vai
ótimo.
ficar tarde. Mas você vai receber uma conexão aí para poder resolver o problema. E eu vou, então, convocar o professor Rubens, da Fiocruz de Minas Gerais, né, que vai falar para nós sobre um método de colorimetric and fluorimetric lamp for species-specific detection of leishmaniasis, ou Leishmania SPP. Então, professor Rubens, com a palavra, tá bom? Isso, ok.
I was muted. So thank you very much. I hope you're seeing my shared screen, right? And can hear me?
Yes, nós estamos lhe ouvindo, Rubens.
Okay, great. So I will speak in English or in Portuguese?
In English.
Okay, great, nice. Thank you very much. First of all, I'd like to thank Dr. Alexandre, Professor Alexandre, who invited me to talk today here in this Tropical Medicine Research Center meeting. It's a pleasure and an honor for me to share a little bit what we're doing in the lab. So I'm a researcher at the Instituto Oswaldo Cruz, which is part of Fundação Oswaldo Cruz, Instituto René Rachou, which is part of Fundação Oswaldo Cruz here in Brazil. And the Instituto René Rachou is located in Belo Horizonte. So many people think that Fiocruz is just in Rio, but we're here based in Belo Horizonte. And it's also an honor for me because I was born in the Northeast. So it's a pleasure to reconnect with people from Ceará. So thank you very much again. Today we're going to talk about, it's a little bit technical. So I will share what we're doing on LAMP, which is the loop-mediated isothermal amplification to detect species-specific DNA from Leishmania parasites. If you're interested in this presentation, I'll invite you to take a picture of this QR code because I just presented this talk in a SBPZ talk, which is part of the Brazilian Society for Parasitology, and these slides are very, very similar. So, okay, when you're talking about diagnostics of parasites, the first thing we think is to see the parasite. So, as you can see here, these are leishmania, and we're sure that they're leishmania because we're seeing the parasite. So, microscopy is very specific, but sometimes this is not sensitive enough to be considered the only, the only technique to be used for the whole leishmaniasis diagnosis. We have also the antibody-based tests. So this is an example of the rapid tests that are being used for COVID-19 to detect the antigens from SARS-CoV-2, which is important for the pandemics. And we have also the molecular tests. And talking about molecular, is always a given RNA or DNA, right? And normally they're more specific, sensitive when you compare to these other, and the idea will be a condition when we have the three platforms available for a parasitic infection disease. And talking about molecular, normally people knows very well the PCR, which stands for polymerase chain reaction. And after pandemics, even non-specialists are now specialized on these kind of techniques, and they can explain why this is better than the rapid test, for example. But normally the PCR, they are centralized in huge laboratories. We need to buy expensive machines and very well-trained personnel. However, we are now facing a technological disruption. For example, we're sending people to space. If you have money, you can go buy to what's the name of the SpaceX, the company from Elon Musk, and you can go and take some pictures of the Earth. And also talking about Elon Musk, we are all connected all over around the world based on his technology that is, improving the communication with the satellite internet-based today. So, and what are these advances have in common? They are allowing us to have access to technological solutions, right? So this is an example of an astronaut, Dr. Peggy, doing a PCR at the International Space Station. And this is a mini PCR, which is a mini thermal cycle, doing high quality PCRs as the same as we're doing in huge laboratories here to detect SARS-CoV-2. So why not to use not only PCR, but also sequencing the DNA in this space? This is also available currently, right? We're using this MinION. Here is the Oxford Nanopore technology. So all are miniaturized and providing us a good quality sequencers and thermal cyclers. So why not to use this kind of point of care technology to apply during a medical appointment? For example, we're talking about leishmaniasis, and this is an example of a dog that was infected, but we can also talk about human disease and use this kind of point of care solutions to the diagnostics. So based on point of care solution, the World Health Organization established this acronym, which is called ASSURED, and each letter comes with a requirement or a ideal solution, a point of care solution. So the A is for affordable by those at risk of infection, sensitive with very few false negatives, specific with very few false positives, user-friendly tests that are simple to perform and require minimal training, rapid to enable treatment at first visit, robust, for example, not requiring refrigerated storage. That will be the ideal condition for sure. Equipment free and environmentally friendly, and also delivered to those who need most. And also recently a group add the R is for real-time connectivity and easy of specimen collection. That are the characteristics, let's say, that are following the technology, right? Real-time connectivity, this is always because we are now under the 4.0 industry and everything is connected. So currently it's easy to have an app or a device that can be connected to a data cloud and send you all this the data. And the ease of specimen collection is normally because for point of care solutions, I think the main drawback or the bottleneck is the pre-analytical phase. I mean, we have to access the sample and we have to pre-treat the sample to be able to run the molecular diagnostics, right? So unlike the PCR, today we're going to talk about the isothermal DNA amplification based on LAMP. There are several techniques that are used for amplified DNA in an isothermal manner. That's the main difference from the PCR technique. And the LAMP use a DNA polymerase that's called a BST. BST is the most common enzyme. A set of 4 primers at minimum that recognize 6 different positions. But normally we're using 6 different primers with a range of temperature around 60 to 65. That's important because if we compare with other techniques here that can amplify non-specific targets at low temperatures, low, let's say 37 degrees, we can consider low in comparison to 60, right? And the reaction time is less than 1 hour. The target's normally the DNA, but however, we can attach the RT or do a transcriptase using RNA and amplify the DNA at the end. And the technique has a high efficiency. So there is a study from a biotech from Germany saying that most of the recent data, we can see more than 65% of these studies are considering the LAMP and the RPA as the techniques that are associated with the new diagnostics tools. And we are raising and increasing here the number of CRISPR-Cas based tests. Here is around 12%. Sometimes the RPA or the LAMP are the techniques that are used to amplify the DNA previously to the detection using the Cas nuclease, CRISPR-associated Cas. So briefly, these are the primers that are used for the for LAMP amplification. So the F3 is the forward or the outer forward primer, and the B3 is the backward also outer primer, and the FIP and BIP are the internal sequences. And after a first step, what we have is this kind of these are the loop. Those are the loops, and the loop primers target exactly this region. So in summary, we have here the non-cyclic first steps, and after that we have a cyclic step and the elongation, which means that we don't have just one band like in PCR reaction, right? If you migrate in a gel, what we have are different bands based on the time that they are elongated. So this is an example of a positive result here. This is a negative and the marker if we migrate in an agarose gel. Talking about primers, today is quite easy to do, to design these primers. There are 2 different tools. The NAB LAMP tool that is supported by New England Biolabs, and the Primer Explorer that is supported by a company in Japan. Actually, the inventors of the techniques are from this company, the Icon Chemical Corporation. And you just put your sequence and you see if you can get the 4 or the 6 different, the 4 and the loop primers to your reaction. And normally we validate the set of primers previously to select the good ones. Okay, in terms of outputs, as you just saw, we can migrate the products on an agarose gel, or we can have, for example, here we have fluorophores, DNA intercalators that we can use under UV or by naked eye. Those are positive, and here is a negative control. We can also measure by turbidimetry, because during the DNA amplification, what we have is a precipitation of a magnesium pyrophosphate, and because of that, we have this kind of precipitation. This is a calcine, so these are positive and negative. pH sensor or ion magnesium sensor. This is HNB. So these are negative and positive samples here with the malachite green. And there are several sensors and markers that we can see by colorimetry. We can also adapt and do the output using real-time PCR machines. So instead of have all the chemistry for RT-PCR, what we have is a LAMP, and we can mark these primers with a specific fluorophore, and we can see the gain here, the fluorescence by time. We can also combine the amplification product using LFD or lateral flow device. And we can see the positive reaction by this test line and the control line. So talking about the LFD, here we have to tag our applicants or the product or of amplification from the lamp. Here is the case with biotin labeled and also fits, which means that after passing in a pad, in the conjugate pad, what they will carry will be, here's the biotin, that there is a streptavidin to take it. And also there is a gold nanoparticle covered with an antibody specific for the fits or anything we have, and this is the specific lane. So here we are combining the amplification, the isothermal amplification, together with the LFD. There is another kind of combination also, not using LFD, but also using gold nanoparticle, in which the gold nanoparticle is functionalized with a specific DNA part that is complementary to our target, which means that in presence of our specific sequence, we can see a stabilization of the gold nanoparticle, and there is no change in color. However, if we don't have the complementation, what we have is an aggregation of this gold nanoparticle, and what we see is an aggregation, and there is a huge difference in absorbance that can be used for point-of-care diagnosis solutions. In terms of pH sensors, during the DNA amplification, we have a decrease in pH, which means that there are some protons that are being released, which means that if we have within your sample a pH sensor after the DNA amplification, we can see here the yellow that are the positive and the pink that are the negative, right? We can also combine the lamp amplification with CRISPR-Cas based. In that case, what we have is a Cas nuclease. And after recognition of the target, the Cas initiates a collateral cleavage, which means that every piece of DNA that is around will be cutted. In that case, the piece of DNA is a fluorophore with a quencher. After the chopping up of this sequence, what we have is a fluorescence emission. In this case, it's Cas13 that targets RNA, but we can also use Cas12 that is targeting the DNA. And we can also combine the Cas-based detection with the dipstick, the LFD, lateral flow device. The lamp using pH as a colorimetric output, it's been used for the pandemics, for the SARS-CoV-2 detection in agriculture, also to detect Wolbachia in mosquitoes, and as a proof of concept, it was sent to space also, and it can work on microgravity as well. There is another very interesting approach, which is called displacing probes. In that case, what we have is here's the loop backward primer, and together with this primer, we have another special sequence here with a fluorophore. And in the reaction, we have the complementary sequence with a quencher, which means that since the enzyme is present, in presence of the target, the enzyme will start the polymerization, and the BST has also an helicase domain, which is able to take out the quencher, and the fluorophore will shine after this recognition. And this is very specific. The fluorescence from a DNA intercalator is quite inspecific, right? Because every DNA you have in the sequence, in the solution, will see a color or you see a fluorescence. In that case, the fluorescence is present only if we have the target. And that was used in 2017 to detect Zika virus, dengue, and chikungunya in a multiplex manner, which is quite nice. In the same tube, you can have all together and detect specifically different targets. Here's the singleplex analysis of chikungunya, and after multiplexing, there is a delay in terms of time. in data acquisition. However, in the multiplex is in presence of all the other primers, and the reaction is going very well. After 20 minutes, we were able to see specifically the virus, and that was used for Zika and for dengue as well, in the same manner. They also did the technique using the mosquitoes. In fact, the Zika virus infected mosquitoes, and the preparation was made in a paper. And after the reaction, we can see here easily the positive mosquitoes. And the main thing, I mean, the main advantage of this is to have in the same tube different targets and identify in a short time of period these different targets we want to differentiate, right? And why this is important for leishmaniasis? Here we have some feline leishmaniasis. I mean, leishmania that infect cats, and this is a species that is dermotropic. I mean, it's causing cutaneous leishmaniasis. And here, what we have is another disease, but it's quite similar, but this is sporotrichosis. And so we can use the lamp using displacing probes to tag the specific primer and see if we have leishmania or if you have other organisms or other pathogens, or even to differentiate species of leishmania parasites. We have recently a case in Sergipe, I think 2 years ago, with a fatal visceral leishmaniasis that was caused by Leishmania infantum. But however, it was not leishmania. In fact, it was critidia. So we can also develop a lamp-based point of care diagnostic system that can differentiate between leishmania and critidia, which is let's say, causing of leishmania. So based on that question, this was a work that was developed by Ellen Gonçalves during her master's 2 years ago. And the main goal here was to develop a tool that was able to differentiate, I mean, to detect leishmania and to differentiate in leishmania species. Okay, so the first step we did was to take different genomes of leishmania and to compare among all these genomes. And that work was made in collaboration with Vinicius from Universidad de Santiago. And what they did was to compare all these genomes, and the sequence that were shared, we're not interested on that. Or we were interested on the sequence that are not, that were not shared, the accessory genome that is called. And here, after this accessory genome, we select some regions that were sent to the tools to design the lamp primers. And after that, we select specific set of primers to detect Leishmania infantum, Leishmania brasiliensis, and Leishmania amazonensis. That are the main etiological agents, brasiliensis and amazonensis, cutaneous leishmaniasis and Leishmania infantum from visceral leishmaniasis. So after that, Ellen, she ran the primers and she selected one of the sets. And here, what we can see is the validation of these primers. Here's the primer that was developed for Leishmania amazonensis with the DNA input of Leishmania amazonensis that were not crossing with brasiliensis, neither Leishmania infantum or the human DNA. And this is the negative control. And the same was seen by her for Leishmania brasiliensis and also for Leishmania infantum. And after that, what she did was to adapt the system for the colorimetric output. And here what we can see is a titration using different concentrations of Leishmania infantum DNA. And we have a limit of detection around 0.01 nanogram, which means 10 picograms, which is quite enough for clinically relevant concentrations of the Leishmania DNA. And this is the confirmation of the amplification. So the yellow here corresponds to a specific amplification. This is the negative control and a concentration that was not able to be seen by the primers. Okay. And she did the same thing, but in that case, mixing with the other primers, the primers that were targeting for Leishmania brasiliensis and Leishmania amazonensis, and the reaction was performed almost with the same colorimetric output. As you can see here, we can easily differentiate between positive and negative samples. So after that, running these tagged primers, so she was able to, after 1 hour, detect specifically Leishmania infantum, amazonensis, and brasiliensis using different fluorophores at the same time, right? So this is the same reaction, different tubes, but same reaction. And what she did after was to mix all these primers, and what she had was a huge delay on detecting, let's say 140 minutes, so more than 2 hours of detection. However, this is inside the same tube, which means that the other primers are impairing the reaction, but we can see here the signals are very specific, and we can differentiate among the Leishmania species that are in the same tube. After that, what she did was a clinical validation. So this is samples from dogs, blood and bone marrow. And as you can see here, she got a 88%, almost 90% of sensitivity. And also for, so these are from dogs, and also for cutaneous leishmaniasis that were caused by Leishmania brasiliensis. In that case, we have less parasites in the lesions, and this is the parts of pieces of lesions from the human skin. And she got almost 60% of sensitivity. And this is a preliminary result, but it's quite interesting, especially for these for Leishmania infantum. After that, Alessandra, she's continued her work, the Ellen's work, and she just started her PhD this year, and she bought new primers with new targets, and now she's improving. And today we're able to see, to differentiate the 3 different species in around 20 minutes. Even for Leishmania amazonensis here is a little bit low in terms of signal, but this is enough to differentiate from the NTC, or the non-template control. She did also a cross-reactivity assay. So in this case, what we're seeing here is the reaction with the primers for Leishmania amazonensis, here reacting specifically for the DNA from Leishmania amazonensis, but not crossing with other Leishmania species. So this assay was made in collaboration with Rodrigo Soares, Jerônimo, and also Felipe. Yes, maybe there is a little bit of cross-reactivity here with Leishmania mexicana, but this is normal because Mexican is very similar in terms of DNA sequence with Leishmania amazonensis, right? So she did the same thing for Leishmania brasiliensis, and here Leishmania guianensis is crossing, reacting. We have a positive reaction here, and it's same results from same case for Leishmania mexicana. Leishmania guianensis is part of the same subgenus of Leishmania brasiliensis, and that's why the sequences are very similar, and this is not a problem. For Leishmania infantum, there is no cross-reactivity with all species that were tested, so which is quite nice. And how can we apply this knowledge to the society? I mean, to translate what we have now and to bring to society with a solution. So our group, together in collaboration with a company here from Belo Horizonte, we developed this device, the OmniLamp, which is a portable device to detect any RNA or DNA from pathogens using LAMP as a technique. So the device is controlled by an app, which is important for to manage the reaction, the LAMP reaction, and also to manage the input of the samples. So as you can see here, we can read the barcode or QR codes. And also the app can detect, can automatically identify the samples based on the color. And as we can see here, we have the test and the control. If they are reagent or not, we see the results very rapidly just after the reaction. And what the OmniLamp does is just to recognize the yellow color as a positive reaction, and the pink will be the negative. And these colors can be adapted. For example, if you're using other sensors, we can adapt the software to recognize the color, which color is the positive one. And a great thing is the device has 12 slots. So 2 slots are for the controls, the positive and the negative for the running. And we have here the couple of samples. So we can have the control and the test sample, or we can have just the samples, which means that we can run in the same batch of time, 5 samples, or we can double that to 10 samples. And what we're doing now is adapting the LAMP reaction and increasing, increasing sensitivity and reduce time of reaction, adapting this system together with nanotechnology. So this work was made by Ana Carolina, Leticia, and Amanda. And the main goal here was to use gold nanoparticles to improve the output of this isothermal amplification. And just to remember, when we have the DNA here, we have this nanoparticle stabilization. So the color will stay like a pink. And if you don't have the LAMP products, I mean, the DNA was not amplified, they will aggregate. And what we have is a difference in signal. So in that case, we are using DLS, which is a dynamic light processing. And as you can see here, this is the time that is used. Here's the volume of LAMP in function of the time. As you can see here, the negative control was still here. Here we have the biosensor, which is a nanogold with a PC of DNA that is complementary to our LAMP product, specifically here for Leishmania infantum. And when we put the amplicon in the reaction here, for example, 20 microliters of the amplicon from LAMP, we have this kind of arrangement, and the light will take a little bit more to stabilize when we compare with the negative or the control. The control is just the nanoparticle without the DNA, right? And this is the same thing for Leishmania amazonensis and for Leishmania brasiliensis. And this is quite nice. What we're doing now is to adapt this DLS system. And this is a real life experience here. As you can see here, this is the negative and the positive. And the changes in using the gold nanoparticles are very sensitive. So after a few minutes of LAMP reaction, I can already see this kind of differences. And we're now trying to put together the LAMP reaction, not the colorimetric, but after the amplification, we'll try to do the YANG, which is the name of the system. That stands for isothermal amplification and gold nanoparticles. So that said, I'd like to thank you again, Alexandre, and the people here from the TMRC. I'd like to acknowledge my students in the lab, the alumni that participate in this project, and our collaborators, and also our partners within Fiocruz, and for sure the funding agencies that are essential to developing science in the world. Thank you very much.
Well, thank you very much, Rubens, for your very nice development of this LAMP diagnosis for an important public health disease, which we need a multiplex LAMP like you have shown that can work. Yeah, that's what I understood. But do you have, I mean, we talk, I mean, Alexandre and you talk a lot, I mean, the last couple months last month actually, because of the FINEP application of the LAMP to the field, you know. So we really excited to test your LAMP developed for the field study in human, like a cohort study, which is the design that we have done for that approach. So I'm very hope that the FINEP will support that. But tell me your expectation for this application. Is that you can help it and you have available? I mean, we do have the support if it comes for the FINEP approval. And we're very excited to have you on board to test that, because we're going to need this approach, because we need to detect case, we need to go forward for the metagenomic and co-infection, etc., which we had been wrote with Anastasio and Adam, who is present here. So comment on this application, because I think you have actually most the application have done was laboratory in vitro sensitivity and specificity. I think the clinical testing is going to be a good approach for you.
Yes, you are right. I agree with you. Thank you very much, Aldo, also for having this session to be a chairperson here. Yes, even without the support from FINEP, we're all open, and I'm especially interested on this project. You can use the lamp we develop here with your samples. I mean, we can do this here. We can send. I mean, can send the reagents to Fortaleza, or we can go to Fortaleza, or people from Fortaleza can come here in Belo Horizonte, and after a couple of days, let's say 2 weeks, I think we can run all the samples. Let's see, I don't know the number of samples, but in 2 weeks, I'm quite sure that we can run a lot of things. Okay. We're not exactly using the, I mean, if you can use the OmniLamp is fine, but I would say when you have a huge batch of samples, normally we do using thermocyclers, or you just put one temperature and go, just because of these slots. It's 96 per hour, right? However, if you need the OmniLamp in the field, for sure that we can go and send the OmniLamp. However, we have to optimize before all the ramp times or the finest stuff that we need to go to the field, because in the field, maybe or not only us that are using the system, right? So we have to train personnel as well. So it takes a little bit more of time. Talking about the maturity of this technology, the OmniLamp is right now being used for the SARS-CoV-2 detection. And that's in the market. So there is a lab that is one of our partners that are doing the diagnostics here. For sure, after the Omicron that is very well working on the rapid tests, the cost to do a LAMP reaction was increased when you compare with the real-time PCR, right? And this is not an advantage to use LAMP now to detect SARS-CoV-2. But depending on the application, I mean, for Leishmania, it's very nice, because in terms of talking about epidemiology, I mean, for clinics, I don't think that's important to differentiate among species, because the veterinary medicine physician will say, okay, this is leishmaniasis, let's go to treat when it's human, okay? Even when it's dog, because now we have meteflosin for dogs in Brazil. However, for example, in Bahia and also here in the north of Minas Gerais state, we're having dogs with visceral leishmaniasis, I mean canine leishmaniasis, that are being caused, or at least Leishmania amazonensis are in the host. And also we have the cutaneous leishmaniasis when you can see Leishmania infantum. So why are these dynamic happening? And this is not new. But now people are like, I mean, looking for that, and they are seeing more strange things like this, right? And that's the case for Sergipe as well. That's why for me, I'm quite interested on that, because in terms of epidemiology, you can differentiate, and maybe you can track and try to answer the dynamics or the physiopathology of leishmaniasis in Brazil.
Excellent. We do have 2 more people to ask questions. I mean, Marco and Adam is going to follow questions for you, okay? Go ahead, okay. Marco.
Hi, I actually have a question that is more like maybe a future research endeavor. I'm not 100% sure if that's going to work, so I would love to hear your opinion on this. We developed this new SARS-CoV test for using LAMP for the detection of SARS-CoV-2 in nasopharyngeal samples. Mm-hmm. And we observed that one of the primers had a specific mutation that actually prohibit the identification of the P1 variant. So I'm actually wondering if we could actually use this kind of mutation, this kind of variation on the genetic code of the pathogen, to differentiate them using a LAMP that is able to identify the whole organism, but one of the LAMP primers, in our example, would be the LB primer, would have degeneration. So for example, for the general SARS-CoV-2, the wild type, we would have a C, and then in the P1, we would have a G. So just this different nucleotide was able to differentiate, let's say, if we had a quencher that has a different fluorescence or detection, we would be able to differentiate the variants of SARS-CoV-2 using the same set of primers. And I'm just not 100% sure if this is like a thing that we could pursue, and I want to hear your opinion on this. What do you think?
Yeah, great. Thank you, Marco. Yes, there are several, I mean, now it's several, because 1 year ago was almost nothing. But yes, people around the world, they are developing new technology or adapting the LAMP system to detect, to specifically detect some mutations. But the first studies are on drug resistance in bacteria. So there are some very nice papers using LAMP to detect these pathogens, and the primers are able to identify one SNP. It's exactly that we're calling, right? For SARS-CoV-2. And yes, there is papers, at least 2, specifically using LAMP and the N region and the S region sometimes, when they use the mutations to design primers and to design specifically a reaction that specifically is targeting this kind of variant in the case of SARS-CoV-2. There are other enzymes and other kind of primers, not only the conventional primers, that we can also use hairpin primers that can engineering a system that can specifically identify a given target. And this is very possible. And you were asking me, are you pursuing on that? I will say it depends always on the target. Because when the stuff is on the paper, sometimes we try to reproduce and it's quite hard to reproduce, right? In terms of SARS-CoV-2, I think it's feasible because the whole world are doing the same thing. So we are just feeling a short communication on LAMP for SARS-CoV-2, saying that even with the mutations that are happening in Omicron variant, we can use the primers that we're using for E and N genes, and there is no problem on positivity. Sometimes people they're a little bit like, how can I say, this is a novelty. However, LAMP is not novel, right? We have techniques at least 20 years. But people are afraid when you're talking about point of care, because sometimes people said, this is not sensitive enough, or there is not specificity enough. However, I would say that this is high quality results. And even the devices like, how can I say, like a toy, it seems like a toy, but open science normally is going to low cost, right? But the results are very, very high quality, and I'm quite confident that the world are moving through the open science.
Yeah.
Yes.
It's actually something that we really talk about in the lab, because the whole work's developed around detecting pathogens here, PCR, even though PCR requires a lot of infrastructure, and third world countries, they don't necessarily have all of that infrastructure. So a LAMP technique would help way much more than a PCR here. But I understand the worry about Europe and the US and other rich countries in changing all their system if they already have something that works. But for us, it will be a lot of help.
Well,
for sure. Yes.
Thank you. Your talk was excellent. Thank you.
Yeah, let me.
Good, thank you.
Yeah, I think it's promising, Mark and Rubens. I think even though if FINEP not come with anything, I think we probably have enough enthusiasm to progress with this collaboration for sure. I'm going to ask Adam to make a short question for you, and you a short answer for us to call Professor Anastasio, who is now, I have his slide on, which I can progress with his talk. Okay, Adam, please. Yeah.
Okay. Hello, everybody. Great presentation, Rubens. I did like a lot your presentation, and the technology actually. When I did master's and doctor, my PhD, I always asked why people are not using LAMP. And I think you have great knowledge of the case of the technology. My question, actually, I have a comment and a question. My comment is that in dogs, we do have visualization of the cutaneous leishmania. So the categorization of the disease are human. I always say that on my presentations. And I do agree with you, for veterinary clinics, it doesn't matter as much to identify the species of leishmania that are causing the disease, because the treatment will be, or the management of the dog will be almost the same. And normally are infantil here in Brazil. And the question actually is, I did see that you're using a LAMP as a hope, or maybe a target should be a point of care method to diagnosis for infectious disease, at least on the first part. But my question is, do you believe that this method will be simple enough or robust enough to be done by the clinic, or will be always on a laboratory of support, or in a big hospital that has the pathologist that does the method? Because sometimes when you think about point of care, and I do agree that LAMP is much more simpler than the conventional PCR, real-time PCR, but still I cannot imagine a clinic, at least a veterinarian, doing a LAMP in the clinic with the dog and the family of the dog over there in the clinic, maybe on the laboratory. But I do want to hear from you what you think about as a point of care test, or as a lower cost, fastest laboratory test. Yes.
Great question. Thank you, Adam, for your words. Yes, I agree with you that I mean, the bottleneck for point of care is the pre-analytical phase. And talking about dogs, we have to take let's say the peripheral blood, and we have to process that, right? However, yes, there are some methods that we can use, like boiling the sample and take this supernatant, which you need a little bit of infrastructure to do that. And we have some solutions already available in the market. There is the M10, for example, that is just a cartridge. When you put your sample in this cartridge, and there is several microfluidics, apparatus in this cartridge. And then we can go for LAMP reaction, or you can go for RT-PCR reaction. And you ask me, is this point of care? I would say yes. However, this is quite expensive yet, right? This is quite, this is like let's say a medium machine in terms of size, but you can have it in your lab. So this is a LAMP and could be considered a point of care. However, I can see that today people are like using synthetic biology and microfluidics to design device, and also with this CAS. I mean, today we have some papers showing amplification free. So maybe we're not talking about the amplification phase, depending on the concentration of your targets, right? Or you can like purify and concentrate your targets to use a technique that is amplification free based on CRISPR-Cas. So after that, we can have everything in a paper, and you just put your drop of your sample, and the paper will change in color. So this is already available in terms of point of care. I mean, I'm in a step from the LAMP right now, but today what you're seeing is these samples need to be amplified previously. So it always depends on the target. This is feasible for SARS-CoV-2, especially the Omicron, because the viral concentrations is high. However, we can adapt for Leishmania diagnosis as well.
Perfect.
So again, thank you very much, all of you, for attending and the questions, and thank you, Aldo and Alexandre, for inviting. Thank you.
Nós que agradecemos, Rubens.
Valeu. Although you're muted.
Boy, yeah. Thank you very much, Hope. Yes, I was telling you this. And let's move on to Professor Anastasio, who I think I have his slide. I think I can share it now, the screen, and I hope it's open.
Yes, it is. Should I begin?
Okay, good. So
Sorry, but even with the specialists, I could not.
tô vendo aqui a tela, tá vendo aí?
Well, our talk is Leishmaniasis, epidemiology and clinical spectrum. Of course, these are very interesting point of the same disease, and we will give like just a little bit of information about these two aspects. I think we should move to the.
Okay. Yeah,
Can you,
okay. Okay.
Can you move to the next slide?
Let me. Vamos ver aqui.
I can't do it.
Yeah, okay. Deixa eu ver aqui. Só um momento. Yeah. Cadê meu microfone?
Tá ligado, seu microfone tá ligado.
Ok. Let me. Eu vou sair e reentrar de novo para ver se realmente coisa.
Mas é só você clicar, não?
Eu vou reentrar de novo porque eu acho que é só isso. Compartilhamento aqui, né?
Sim.
Ok. Acho que agora tá. Dá para passar. Isso, beleza. Você me avisa aí que eu dou o toque aqui, tá bom? Vem aqui. Cadê meu microfone? Ah, tá aqui em cima. Ok. Vem aqui.
Let's go to the next slide. I'm sorry,
okay.
My microphone was. Okay, this is a slide showing the many species that cause leishmaniasis in human. We have listed here 20 species. All of them cause leishmaniasis in human. In Brazil, we have infantum causing visceral leishmaniasis, sometimes cutaneous leishmaniasis. Brazilians, Guianense, Panamense, Shawi, Naif, Langston, Lindenberg, Peruviana, Colombiana, and that's all. And they cause, you see that the infantum cause cutaneous disease, and Brazilians and Panamense, besides cutaneous disease, they cause also mucosal disease. Let's go to the next slide. This is a very interesting slide. It's on visceral leishmaniasis. Just to give you an idea, how many cases are not reported, even with a very serious disease. We have a great difference. This is a calculation made by WHO. See that we report 3,000. Let's not consider the year. We report 3,400 cases, and it could be from 4,000 to 6,000 cases. So we may not be reporting thousands of cases. That's with visceral leishmaniasis. Let's go to the next slide. Concerning cutaneous leishmaniasis, the problem is much bigger. It's much concerning. See, we report, let's say, in this year, 26,000 cases, and at least we were reporting in the best of the hypothesis, we were reporting 1/3 of the cases. We may just reporting 1/4 of the case. So many cases of cutaneous leishmaniasis are not reported for many reasons that we are not going to discuss. Just to give you an idea that all disease in our great country and many other countries do not have a very correct reporting system. Let's go to the next. This is just to illustrate your case of visceral leishmaniasis. See, in the Americas, most of cases, if you see in red, it's the Americas, and in green in Brazil. Most cases are in Brazil. The other countries have very small number of cases, just to illustrate that visceral leishmaniasis in the Americas is a problem in Brazil. It's a huge and important problem in Brazil. Let's go to the next. The next. Okay, that's cutaneous. You see that cutaneous, it changed a little bit. The area in this dark blue over here is Brazil, and the red is the American. So you see that many countries like Peru, Bolivia, and several other, Colombia, they have many cases. So but even though almost half of the case of leishmaniasis in the Americas are in Brazil, of course. Let's go to the next. This is just to give an idea, even though a disease that's about the number of deaths in visceral leishmaniasis. See, the percentage of case that people die is very high. See, in 2012, it was about 6.8%. And in the last year, it increased. What's the reason? Is the health system? Maybe not, because we have many immune-compromised patients as well as old patients. Old patients, sometimes they took very long to have the diagnosis made because as they have anemia, sometimes they do not have fever, so they take very long to have a diagnosis made when they are very sick and do not improve with treatment and die. That's a big problem. But we have to see if the health system is not prepared to diagnose this case, because if you analyze the health system, because it's a disease easy to diagnose and there is very effective treatment. Let's go to the next. This is just to give an idea, the percentage of people with co-infection of leishmaniasis. See, in Paraguay, it's very high. In Brazil, it's about 10%. Of course, this from 2016, but it probably didn't change much. From every 100 cases of leishmaniasis, at least 10 cases are co-infected people. So it's very important to have this co-infection diagnosis made, because the follow-up, the way to treat this patient is different. The time of treatment, it's much longer than in the normal or a non-immune compromised person. Next, please. Of course, when we talk about leishmaniasis, we always think about the vector. Is a vector-borne disease. Is a disease caused by a vector. But in fact, there are other ways of having the disease transmitted. Let's go to the next. This is dog, because dogs are in fact the mosquito, and the mosquito transmitted disease. The next. This is the work many years ago made in Spain, published by Alvar and co-workers, where you see that drug addicts, as they share needles with people, in fact, with leishmania, the blood that the remainder of blood that stains the needle when they inject. So, because this disease, people have parasitemia. Even a very low parasitemia is enough to contaminate the needle and permit the transmission to other people. So even though it's caused by the mosquito bite, it's zoonotic endemic leishmaniasis, you have the anthroponotic epidemic leishmaniasis. Of course, probably in all places that we have many drug addicts, you have this type of transmission. The next, please. We'll see this world map where we have very dark red. As you go from very dark red to to a less strong red, you have a smaller number of species species of Leishmania circulating in that country. See, the Brazil has eight species of Leishmania that circulate and cause disease in men. Of course, most of them in the Amazon region. In Ceará, causing disease, we have just two: Leishmania infantum, that it was called Chagas, and Leishmania viana brasiliensis, that cause cutaneous disease. Of course, Leishmania amazonensis, that has been well described in Bahia, its cases are very rare, very rare. We've never seen a case in Ceará, even though this discussion has been. So in the yellow, in the yellow, you see there is no number, no letter. So in this country, Leishmania have not been reported. In some other country, like you have just one species. In the United States, I will not present, but the Texas is an endemic region for cutaneous leishmaniasis. In a recent paper, there are 45 cases autochthonous from Texas. It was published, I think, in JAMA Dermatology. I didn't bring it here. So just to show you that it's a very important disease, very disseminated. See, in Africa, almost all countries have the disease, and in Europe, in the Middle East, and Asia, and in South Central, and North America, excluding only Canada. Let's go to the next. This is just to illustrate what I just said. It lists all the species. South America, we have many species in different countries. Africa, we have 1, 2, 3, 4, 5 species in Africa. And you have several species also in Asia. In Europe, you have 3 species, but the importance is infantum, causing visceral leishmaniasis, sometimes cutaneous, and tropica, causing cutaneous disease. So it gives an idea how many species circulate in each region. Next, please. This is just to illustrate. It's a paper from Parro, but I just want to illustrate. I'll show here Peru and Ecuador. See the number of Leishmania. You see Leishmania brasiliensis. The green is Leishmania peruviana, and Leishmania guianensis. So just to illustrate that in the same countries, you have several species circulating, causing disease. Of course, in these countries, the most important species causing severe disease, especially mucosal, is Leishmania viana brasiliensis. Next, please. This is just as a node map, but it just represents almost what is happening today. It didn't change much. See, the very dark red areas represent the areas with lots of cases of Leishmaniasis. In Ceará, just to illustrate, in the north, all the region, Meruoca, Tianguá, all those Serra da, all those places, we have many cases. But Ceará, the disease is disseminated in many, even Fortaleza, there are autochthonous cases. We have seen people that they swear that in the last 5 years, I did not leave Fortaleza. So if they get Leishmaniasis, it's because it was. And the vector, when people try to investigate the vectors that cause visceral Leishmaniasis in certain section of Fortaleza, they've been capturing many species, several species of cutaneous, like migoni, like witchmani, that are Lutzomia litimoni, witchmani, Lutzomia migoni, that cause cutaneous disease, and they do exist in Fortaleza. Please, next. This is just to illustrate the cutaneous leishmaniasis in Brazil. I'm not going through. See, the Amazon region is where the region most affected by leishmaniasis. But Ceará, see almost the whole state with some very few areas that cases are not reported, but we especially the south and the north of Ceará, where most of disease have occurred. In Fortaleza, like Eusébio, Aquiraz, we have seen many cases of cutaneous disease. Unhappily, both diseases are endemic in Fortaleza, mostly visceral in Fortaleza. Please, next. This is just to illustrate the trend from 2001 and 2017. See, at some point, we had lots of cases in Mato Grosso, South Mato Grosso, and but the disease has always been present in the northeast and part of the Amazon region, like Maranhão, Pará, and as well as in Bahia and Minas Gerais. Minas Gerais today is a very endemic state for visceral leishmaniasis in high blood. In Belo Horizonte and Fortaleza, they concentrate many cases of visceral leishmaniasis. Today it is a urban disease. This slide it's a correlation between the incidence of canine leishmaniasis and human cases. So if you have many canine leishmaniasis cases, you have many because the vector contaminates from feeding on dogs and taking the disease, transmitted the disease to humans. So that's a big problem. Next, please. Next, please. Oh, in the past, okay. Well, this is just to show what it was where the case of visceral leishmaniasis were concentrated. Today, they represent few cases. Most cases are in the urban area. Please, the next, please. The next. This is just to illustrate. It's a study done by Tom Evan, we also participate, like Dick Pierce and many other people, Jane, etc. But of visceral leishmaniasis in Northeast Brazil. You see, we studied a group of children, I think was about more than 700, and from these children, during 2 years, 108 children, zero converted. And of these children, only 11% got sick. And a follow-up 10 years later did not show any new cases. Of course, if these people develop immune suppression, receive a transplant, or develop HIV or contract HIV infection, they might develop. So most of people infected with Leishmania infantum, they do not develop disease, but we believe that many of them become with latent infection for the rest of their life. And at some point, if they are immune suppressed, they might develop the disease. The next, please. This is a study done, I participate, but there was Daniela Monteiro, Professora Margarida Pompeu, and many other people. Blood from donors, blood that would be given to people as transfusion, the blood was analyzed, and in those 431 samples, more than almost 13% of the cases had serology positive for leishmaniasis, and 4.8% were PCR positive. What means that probably it's a hypothesis, some of the blood that we give people to help them recover from the anemia or from other problems, they might get leishmaniasis. It's a possibility very strong, but the title was a little bit different, but they changed to not to alarm people. But Leishmania infantum infection in blood donor, Northeast Brazil, was done in Fortaleza, material from the hemolysis. So I believe that people that receive transfusion, they have a very tiny chance of getting visceral leishmaniasis from the transfusion, unhappily. Go ahead. This is just to show that the humans are also reservoir. If you see, for instance, the skin of a patient with AIDS, you do a biopsy on a patient with AIDS with visceral leishmaniasis, the amount of leishmania on the skin is just amazing. So if they suffer a mosquito bite, if a mosquito feeds from that skin, the blood, they will be much more infected than feeding on a dog. So, and Dean in the 1950s showed that humans are, with visceral leishmaniasis, they infect the vector. So that's just to emphasize this point. Let's go to the next. The clinical spectrum. Of course, the number of presentations are very, very large. Let's go to the next. Go very quick. Every one leishmania has some typical presentation, but when you see many cases of the disease, you see all type of presentation. It looks like Leishmania tropica, Leishmania major, Leishmania epiphanoi. The only presentation that we do not see is diffuse cutaneous leishmaniasis, that only in the America, only Leishmania amazonensis cause. And Leishmania ethiopica in the old world also cause Leishmania, we call anergic or diffuse cutaneous leishmaniasis. People have many lesions, no one is ulcerated, and when we do a biopsy, the amount of parasite is just amazing. So, and there is no cure. You give medication, people improve, then they relapse. Let's go to the next. This is an old article, Dick Pearson, where I was great. He is a great man, a great professor, and a great researcher. We had a paper, we published last year. So I started working with this in 1982, and so it's 40 years of work together. So state-of-the-art clinical spectrum is it's a very interesting article, just because we're going to talk about this. I just show a few pictures. This article has discussed many aspects, immunology, etc., and some other aspects of it, but just the clinical. Next, please. We have 4 pictures in this slide. This is a girl with visceral leishmaniasis, a man with localized cutaneous leishmaniasis, a severe lesion on the nose from mucosal leishmaniasis in the lips. And this last picture was from a patient that Pearson cared in the United States, where the patient had visceral leishmaniasis and had pleural effusion, a aspirate of the pleural effusion. It was centrifuge, and one macrophage was seen with, I counted 120 amastigote. So showing that HIV and Leishmania are a very negative clash. When they meet, they really cause lots of problems for the patient. Let's go to the next. Visceral leishmaniasis, people have fever, weight loss, malaise, loss of appetite, pallor, enlargement of the abdomen due to hepatosplenomegaly, edema, diarrhea. Concerning fever, many old people and many people with HIV, they have no fever. Some people with HIV, they feel nothing. It's amazing. If they do duodenal biopsy, thousands of parasites in the duodenum, they feel nothing. It just, they don't feel malaise. They don't have fever. Why? Because who causes these symptoms are the immune system. The immune system is, as I would say, paralyzed. They do not react. So they permit the growing of the leishmania, and the leishmania growing permit the growing HIV. So it's a very bad co-infection. Let's go to the next. These are picked from São José. At some point, today we have much less, but we had in this day, we had 3 other patients that were in the ward, could not come to the picture. We have most of patients today are adult or adolescent. So because the reason why they call leishmania phantom, because it was first described in Mediterranean region, and all the cases were in children. That's why the name. And we still have many children with disease, very advanced cases. Unhappily, this does happen. Next, please. This is an old pic from Sobral, Dom José Tupinambá, Tomás Aragão, and I can't remember the name of this gentleman, was a pharmacist, a great man. So all children, see, we have an epidemic of leishmania in those days, and Professor Alencar was one of the people that studied the disease over there for very long. Let's go to the next. This an old slide of a boy with huge liver, huge spleen. Unhappily, he died because he contracted measles. Measles is a very serious disease for someone with a parasitic one. So when you put both together in a malnourished children, it's just very bad. Let's go to the next. I said edema, see the edema, hepatosplenomegaly, thus little go. The albumin was less than 2, So the anemia gives this pallor, and the low albumin gives this edema that's happily recovered with treatment. The next, please. This lady, of course, she had fever. She had all the hepatosplenomegaly. See, the problem is the only thing that she liked to eat was rice. And see the edema, she had 1.8 grams of albumin. Once she was treated, she became very, very thin but healthy. Next, please. One condition, post-Calazar dermal leishmaniasis. You treat a patient, and after treatment, they appear with this lesion. Does this disease occur with leishmania phantom? No. We have a case of leishmania phantom with huge lesions because the patient had a hyper IgE syndrome, and you treat the patient, the patient recovered, and later on he developed skin lesion, but bone marrow has no parasite. So the disease is a cutaneous form of disease that appears after treatment for visceral leishmaniasis. It's mostly described by Leishmania donovani, not by Leishmania for Leishmania infect. Next, please. Cutaneous leishmaniasis, let's go. I think I was cutaneous leishmaniasis. You have localized cutaneous leishmaniasis. You have Leishmania lymphadenitis that appear before the lesion or with the lesion. 50% of the people with cutaneous leishmaniasis at the beginning, they have lymphadenitis. You are going to see disseminated cutaneous leishmaniasis, diffuse annular cutaneous leishmaniasis. We do not have it in Ceará, but we have in Brazil. I think I have maybe I have a picture. And mucosal leishmaniasis, and I present one with HIV. Let's go to the next. This is bubonic leishmaniasis. It's we with people from Harvard University. Jay Maguire is still working very hard. John David, he retired, but was a very interesting. We worked in Baturitá during several years, and I observed in São José case of people with lymphadenopathy even before the lesion, and we did a very interesting study. But this study, it's a very huge study. It talks about epidemiology, all the parasites, culture, histology, immunology. It's so big, the study, that it's even difficult to read. Let's go to the next. And we call it bubonic leishmaniasis because the node was so big. See, or the lesion, as the lesion enlarged, the lymphadenopathy disappear. Another lesion, see, but this little boy, the gentleman had a lesion in the hand. When I palpate the axilla, had this big, see, and a lesion, the lymphangitis, very common. And if you biopsy this lymphatic, you will find leishmania. Let's go to the next. See, repeat, see, and many of the nodes are mostly femoral, but a little bit of inguinal too. You see, all place. This is from the man with the lymphangitis that I just showed. Let's go to the next. See, the lesion is very small, but you have a huge, and the best way to make the diagnosis is to aspirate the node and send it for culture or to the PCR today. See, big. Let's go to the next. To the next, please. To the next, please. All these pictures are mine. See, no, go back, please. See, the lesion huge and the lymphangitis, see, the lymphangitis. And with the treatment, the scar, as time goes by, the scar become much faint and much, the skin will be a little bit less dark than the normal skin. Let's go to the next. It's very important not to do the debridement, because you let the lesion, you let the crust, it will drop as treatment is effective. You have almost no scar. See, next, please. You see all types of lesions. See, in the hand, in the close to the knee, in the eyelid, in the close to the genitalia. So you see lesions in all places. Let's go. All body part. Just to illustrate, this is a typical lesion. This is a little bit different. So 2 lesions here, another 1, 2 lesions. Let's go to the next. This is a very initial lesion. If you look the eye, there is edema over here. The lymphatics here are involved, and the node in the right mandible, you palpate them, very huge lymph node, because the draining lymph node are on the right, and the edema is see, if you compare, is because of involvement of lymphatic. The next, please. Almost finishing. You see all kinds of things from Mulungu, this lady, just unbelievable. The next, please. See, a very tiny lesion. When we take a picture, closer picture, you see that this is typical leishmaniasis. As a rule, this lesion has at the most a week or 10 days. This is a lesion of 3 to 5 weeks, but it's typical. The lesion is a pizza-like lesion. You have the raised borders with the ulcer. You have 2 lesions that migrate. They are almost together. Let's go to the next. Mucosal leishmaniasis. Mucosal leishmaniasis is a consequence of the parasitemia that occur early in the disease. As I showed, the lymphadenopathy, during the lymphadenopathy, if you examine the patient with detail, you'll find splenomegaly, and in 3 patients, we were able to isolate from the buffy coat Leishmania brasiliensis, showing that the Leishmania circulate. When the Leishmania circulate, they become trapped in the cartilage of the nose, and there it stays latent for after many years of latency. For some reason, they start causing disease. You see this, the same gentleman, if you look over here, they have involvement of the upper lip and the nose. When you look the nose, all the septum is destroyed. This, it comes to this point after at least 10 years of evolution, or 7 years, not less than that. It takes, see, the patient have lots of difficulty to have a clear or a valuable or a qualified assistance. You see the Montenegro in this patient, 40 to 15. So you have 25 to a Montenegro test of 25, and there is an area of a necrosis. Later on, it had a necrotic area in the very center, showing that this patient, they have a very strong immunity. They have a very strong response to Leishmania antigen. Let's go to the next. It's interesting because if you look the patient, of course, he has the nose involved. It didn't drop it, but you have the septum. The septum. You put the light one side and you see the other side. The septum is perforated, and some people after treatment, they scar, bring lots of problem because this lady, the nose was not like that, but after treatment, the fibrosis caused this very ugly appearance. Of course, she needed a plastic surgery. She didn't have, of course, and to do a plastic surgery, you have to wait at least 1 year without relapse, so you may recommend a surgery, but this lady didn't have it, so it's very sad. Let's go to the next. So disseminated cutaneous leishmaniasis, it's not a common disease, but we working mostly with people from Mulungu, Baturité, and some other region, we were able, this was published in September last year, disseminated cutaneous leishmaniasis and alcohol misuse. We went the word alcohol abuse, but the reviewer didn't want it. So anyway, what we saw from 18 patients, 14 were heavy drinkers, and we hypothesized that the alcohol use did diminish the response, the immunologic response to Leishmania. That's why they have the disseminated disease. The disseminated disease is when the people have more than 10 lesions in more than 2 body parts. Let's go to the next. See, if all the people that drank less drank 45 grams. So all mostly cachaça, né? Because cachaça and vodka, tequila, and whiskey, they have almost the same concentration of alcohol, 40%. So 800 grams of cachaça means 2 liters per day, 2 liters per day. And you just, so here it's almost 20 is 50, 100, 150, almost 150 per day of cachaça. But most people drink very much, almost 1 liter, see? That's why we believe they diminished their immunity, and the dissemination of Leishmania was favored. Let's go to the next. These are the pictures that we present in the paper. See, in the genitalia, the penis over here, and this gentleman had this horny lesions. See, this gentleman had a like zoosteriform lesion involving a ulcer, so it's very thin. Let's go to the next. This is what it's available from free, because the emerging infectious disease, you can access all the article free of charge. So this 36 gentleman, 6-month history, he had a 167 lesion all over the body, but on the face, the lesion were very strange, very strange. It's unbelievable, but when he was treated and cured, his face became almost normal with no scar. Just interesting. Let's go to the next. This is another disseminated cutaneous leishmaniasis. The lesions are not caused by mosquito bite. It's dissemination by lymphatic or blood. We believe blood dissemination from the parasitemia. And because nobody can have this gentleman have 747 lesions, if I'm not wrong. Let's go to the next. Another gentleman, see? Next, please. Other two gentlemen, see lesion in the eyelid, see? Next, please. Just to illustrate, you use a flashlight to see if the patient has perforation of the septum, because sometimes the nose look okay, but when you use the flashlight, you see that the septum was involved. Let's go to the next. This is a diffuse cutaneous leishmaniasis. This picture is not mine, of course. I never had a case because we do not have Leishmania amazonensis in Ceará. Bahia has described some cases, very few cases, but it seems that disease disappeared. It's a rare disease even in the Amazon region. Not all the people infected by amazonensis do not develop this syndrome, but those that develop, we were never able to cure them. So the disease goes back and forth, wax and waning course with good treatment, with liposomal amphotericin B. The next, please. This is our last slide. The gentleman that he was a veterinary student. He had leishmaniasis, cutaneous leishmaniasis, and the lesion was over here, where I'm pointing, and he was treated, and the lesion healed. A little bit, 1 year after that, he started presenting this lesion in the body, and serology for HIV was positive. We thought initially he had Kaposi, sarcoma, but all these lesions were full of leishmania. Unbelievable. This was Leishmania viana brasiliensis, and he never recovered. Those days, we did not have liposomal amphotericin B. We did not have good treatment for HIV. I think he was before the AZT era. So that's big, very sad. So he died, but he had disseminated cutaneous leishmaniasis. It's not it's disseminated cutaneous leishmaniasis, but almost diffuse cutaneous leishmaniasis without those nodular lesions, but the whole body was involved. We did blood culture several, I think 3 times, all the culture were positive for Leishmania viana brasiliensis. So he had a very severe disease, and he died of the cutaneous leishmaniasis caused by L. brasiliensis. The next, please. I think it's the last. This is when I used to do spleen aspiration for the diagnosis of Calazar, but every time I was very anxious, and happily no patient died in my hand, but I stopped doing it because, but it's very interesting. You take a tiny drop of blood in a full leishmania. Thank you very, very much.
Well, thank you, Anajat, for this excellent view of leishmaniasis in Brazil. I mean, it's a very nice overview. I mean, never seen in the last decades. Thank you very much. And the clinical presentation, it's amazing I know you are a master in documentation, pictures and photos. So you, beside this, you have a good science, so makes the whole difference. I mean, when we talk about disease in presentation like this. I mean, now I think we open for some questions, and let me see my shot here because I probably can see. Yeah, I'm going to say excellent, Professor Anastácio. And so Anastácio is, I mean, probably the next talk, Adam is going to talk about co-infection with canines, like you said, dogs. And do you see there is an explosive combination, like you said, HIV and measles, but do you see any, I mean, how do you see in terms of prevalence of co-infection? I mean, at the primary or outpatient clinics, let's say.
Well,
I know inpatient may be more, but outpatient clinics, I really would like to know about your opinion. Yeah.
No,
go ahead.
well, I understand that in dogs you have co-infection with other microorganisms. In patients with HIV, the protocol for treatment was changed because this is not correct. People used to treat HIV with visceral leishmaniasis, HIV patient, with the same dosages and the same duration as people that not immune compromised, but every patient with visceral leishmaniasis is immune compromised, but not with a clear immune deficiency, like transplant patient, HIV patient. And I think it's a big problem today because dogs and cats that are reservoir of leishmaniasis, especially both leishmania. In Baturité study was done, showed that the cutaneous leishmaniasis, dogs over there the dogs are important reservoir for cutaneous leishmaniasis. In Fortaleza, in everywhere, dogs are very important reservoir for visceral leishmaniasis. So today, as we all know, we do not sacrifice. We do not sacrifice. We do not take them for euthanasia, the dogs. So what happened? People do not kill the dogs, and people have no condition to buy the collar, because the collar, as they have the collar have a insecticide, and the insecticide in the collar it disseminates on the skin and the fat tissue of the dog, and it do not permit the biting, or if the mosquito bites, it dies. So the dog that you wear a collar, it will prevent the transmission of the disease. But the collar is very expensive, and the public service do not give people that have the dog the collar. And so you can't kill the dog, and you can't prevent. So, and you can't treat, because there is no good treatment for dogs, and it's the discussion will be very long. So I think we are in a very sad moment because we have more people immune suppressed, not only from HIV, but from transplant, liver transplant, heart transplant, kidney transplant, etc. So it's a big problem.
Yeah. Well, I'm checking the chat. Everybody say thank you all for sharing so much knowledge. I'm happy and grateful. So that's a good compliment. So excellent. So let's move on. Thank you.
Thank you very much, Aldo and everybody.
Yeah, and we're going for the next one. Adam, if you can share the screen, and take you on. Adam is a recent PhD from State University of Ceará, and now work for the laboratory called Abbott. Yeah, he's doing some clinical and diagnostic coordination for the Abbott laboratory right now. Go ahead, Adam.
Okay, hi to you all. Thank you for the invitation, Dr. Lima, and thank you for reminding my name. It's an honor to be next to Dr. Anastasio and now Dr. Rubens. Hi to Richard Garant. I don't know if Sean's still there, but hi to Professor Alexandre. We are, okay, hi to you all. It's very pleasure to be here with you. So I tried to make a compilation or a short presentation of my data from my PhD. I cannot see you when I'm presenting, so I ask you to say if everything is okay from there. So my presentation is in Portuguese, but I will try to present on English. So our title of our study, I did present it on TMRC on 2020. So who else were there? We'll see that we have much more analytical data analysis on the data. So we will have here a discussion. I did try to shrink down the presentations because I knew that we were on the further part of the day, so many people will be maybe tired, but then we will be open for questions. So I hope it works. So the title is Leishmaniose and Ehrlichiose, canine Leishmaniose and Ehrlichiose in the family profile as risk factors on Fortaleza, Ceará. So we tried to analyze the risk factors for visceral canine leishmaniasis. So Anastasio did a great presentation. I'm glad that I removed some slides from my presentation because I had some same graphs. So the great reason or rationale here was to say that dogs are related to humans. We know that dogs were the first domesticated animal on the story of human. So we do have that phrase that dogs are the best friend of humans, and that's historically and scientifically true. Dogs share the most common disease with humans. They share the most homes with humans. So we do have this intrinsic interspecies relationship. So as we know, Brazil is endemic for several vector transmitted disease. And this is because our climate favors the vectors. So, and leishmaniosis and ehrlichiosis are two diseases that are transmitted by vectors. Leishmaniosis, as Anastasio explained very well, by the phlebotomy, and ehrlichiosis by the tick. It's this one. We will talk about canine ehrlichiosis. It's Ehrlichia canis. So it's a very specific tick. We have a slide to share with you that. So the prevention measures and control for visceral leishmaniasis. I think you all read the program from Brazil, but it should be based or should be rational or should be, let's say, based on reducing the load of the pathogen in the environment to diminish the transmissibility and to try to limit the appearance of new pathogens. So this would be the three pillars that we should aim for. So we should study how the disease happens so we can aim the right pillars of the disease. So this is a new approach of diseases. I think you all read something about One Health somewhere. So as Anastasio said, he said that cats can be reservoirs, humans can be reservoirs, canine can be reservoirs. These are the three species that we are looking at it, but we have several more other species in the environment here in our cities or in rural environment. So we do know today that we need One Health approach. Dr. Lima has a great project on bats with Marco and Professor Alexandre. We do know that we need to monitor environment and other species to attain better health for everybody, not only humans, but for our nature and for our species, whether it's domestic or just living with us in our cities. So this is my figure about the species. I'll jump that because Anastasia was very great of saying about explaining that. And this is a table of paper that we have published on Brazilian journal, Brazilian vet journal, it's PubVet. So the thing here is just to illustrate that we have a lot of cities with this is seropositivity for dogs, and we have a lot of this is the first authors, and these are the states that we have the data from. So the incidence or frequency, depending on the paper, will vary from very low rates to very high rates, depending on the methods that they collect the data. But as we know, in endemic urban areas, we have 5 to 20% of visceral leishmaniasis, canine visceral leishmaniasis positivity. So this is where we aim the study to analyze how important is dogs' visceral leishmaniasis. And Anastasia showed that graph that I think I have it here. It will be on the others presentation. So this is just to illustrate that visceral leishmaniasis is presented, it's endemic in many cities, and it's important because it's a reservoir for human. And as Anastasia shown, we have a lot of paper that shows that the incidence of dogs' visceral leishmaniasis are close related to humans' visceral leishmaniasis. So the incidence normally goes together. We do think that is the dog's fault, but maybe it will be the vector's fault to have this interaction. But the thing is that we cannot decline or deny is that these diseases, human and visceral and dogs' visceral leishmaniasis, are related. This is fact, and if you don't acknowledge that, you will be just closing your eyes to the truth. So this is my figure from the cycle. So just to remind you that we have a vector, it's a biological vector, so it has a parasite face on the intestine of the vector. It is pretty important that this happens for the parasite. And we have a host inside face of the pathogen. But one thing that normally the scientist doesn't account for, or at least epidemiologists doesn't account for when they're making the program for controlling leishmaniasis, is that dogs can transmit leishmaniasis through sex transmission. Males can transmit to females. The inverse way is not proven. As the papers goes and the test goes, I do believe that this not happens from females to males, but males transmit to every female that has. So when you have a male that doesn't have any clinical signs, but it's out the street, he is transmitted by the phlebotomy, and he is transmitted by the sexual act with females. And then what we do know as well is that females transmit to their puppies. So when you have in the United States, when I made some research over there, we do know that this transmission is completely independent from the phlebotomy, and it's completely successful of maintaining the disease on the environment. So even when you don't have the phlebotomy, you can have the endemicity, I can say that, in an environment just with the sexual and vertical transmission. So this is the clinical signs from leishmaniasis. You have from canine visceral leishmaniasis, you have dog here positive but without clinical signs. You have this dog here that doesn't have much clinical signs but has this dermal presentation. This is visceral leishmaniasis. So as I comment on the Ruben presentation, so we may have leishmanian phantom going to the skin of the dog, and we may have cutaneous leishmaniasis going for visceral in the dog, so in the canine species. This is a dog with severe loss of muscular mass. So you have this just bone here, the cranium, the dog going with this very big format here on this occipital, conjunctivitis here on one side, The skin are very good. You can see this is a very typical like bug description of visceral leishmaniasis in the dog. You can see here the big abdomen distension and a low mass here on the legs. So and it's a dog that it has that sad face, that it's pretty tired. And this is a dog that is euthanized on the Central Control Zoonosis on my master's degree. So you have the nail growth, you have the skin all with all the kind of lesions, the point of ear lesions. The eye here, it's completely with conjunctivitis and other aspects of the dog completely with all these clinical sides. So we can look at canine visceral leishmaniasis on a three way. We do have the public health like observation point. So it's kind of here in the TMRC are the it's the biggest, like it's the more important here for our audience here. But you can see the dog, infected dog with Leishmania infantum as a clinical. So this dog will be an individual. It doesn't matter how hard he is transmitting or how less. It does here on the public health, it matters if it's transmitting or not, if it's going to live long with that disease, if it's going to transmit a lot on a short period of time, or if it's going to transmit a lot on a longer period of time. In a clinical perspective, it doesn't matter the transmission, it does matter how the dog is for the family, if the family can live with that dog for several years, and if the dogs can live with a good state of life for several years. So here, as a clinic on a veterinary issue, you should care for the transmission, but on a clinical perspective, you are going for the clinical signs in giving the dog and the family a good life after infection. And as a parasitologist, here will be more for scientists, for us to see if the dog can become negative or positive, if the dog can be clinical health, but has the parasite inside of him. So it's kind of a perspective as you go for the parasite. You're looking for the parasite. It doesn't matter much of the public health or the health of the individual infected. So those three perspectives are kind of related to the three main epidemiological aspects of the visceral leishmaniasis. So you do have the mammals that leishmania can infect. Anastasia was very clear on that. We do have several mammals that can be reservoirs. We do have bats that are infected. Cats are infected, but the reservoir potential are discussed, as we do know that susceptible animals are different from reservoirs. These definitions, it's a balance of the how much it transmits the disease. So this is the vector and these are the parasite, and all of these individuals or all of these items live on the same environment. So the environment must be healthy to inhibit this vector growth, and we should care for lowering the parasite load on the environment. So this we accomplish with treatment of the patient, with early detection. So it goes with the lump presentation of Rubens, and goes with the Anastasia saying that people get infection with leishmaniasis. Leishmaniasis is a very smart parasite, if you can allow me to say that, because it doesn't kill the host very fast. So it does take time to the person to go to search for a veterinary or a doctor to get this diagnosis. So normally it's too late. Normally this parasite goes on the environment already. So we need to care about early detection. And it's not about early detection, like Anastasia said, that it's very easy to diagnose leishmania, and it is, but it's not very easy for those clinicians, veterinarian doctors that are not aware of it. So it's easy for me, it's easy for Anastasia, and it's easy for most of people that are listening to this presentation. but diagnosing leishmaniasis is not easy when you're not accounting for. So we need to get these methods of diagnosis and get these people to get access to that methods. And even if it's a low fever, but if you need to test it out to get this lowering the parasite load on the environment. So ehrlichiosis is a simpler disease because it's caused by a bacteria. It's a gram-negative bacteria. Here on this presentation, we will account for Ehrlichia canis. It's very endemic in our country. The darkest states are more endemic. So we have a lot in Ceará, we have in São Paulo, Mato Grosso, Minas. We have a lot of Ehrlichia in Brazil. Ehrlichia has somebody doesn't know, just to remind, it goes inside a phagocytic cell. In this case, normally it goes for macrophages and it replicates inside a macrophage. So here we can see several morulae of Ehrlichia canis inside a macrophage. We cannot say it's Ehrlichia canis, but we do believe that it's canis because it's inside a macrophage in a dog in an endemic area for Ehrlichia canis. But the morulae itself we cannot distinguish from other Ehrlichia species. So this is the tick that transmits Ehrlichia. It is Rhipicephalus sanguineus. So it is. We do say that it's carrapato de 3 estágios, né? Carrapato de três hospedeiros, 3 hosts tick. So it lives on the environment. It doesn't live on the dog. So when you see a lot of ticks on the dog, it does have much more ticks on the environment because it just goes for the dogs to feed. So it bursts from the egg, goes for larvae, feeds on the dog, goes in the environment, feeds again as a nymph, and then goes to the environment, feeds again as an adult. So, and the females can put almost 2,000 eggs when they are mature and have the eggs. The males can transmit between feeding because they can feed several times when they are adults. The females only feed 1 time because then they put their eggs on the environment and then die. We do have 3 clinical presentations for ehrlichiosis, and these I will focus here on the subclinic. As Anastasia said, the latent patients are the worst for public health because they keep spreading here is the bacteria. On the Leishmania case is the parasite. So here we have the most problematic patients for public health or One Health approach. Here on chronic and acute phase, normally you will have this dog going to the clinic or searching for a veterinary assistant. So these both dogs are not very problematic for public health. These chronic phase are very problematic for the dog, but not the case here in that discussion. So here are the other graphs that shows virtual leishmaniasis growing together for canines and humans. We do have several papers that says that. I will not take long here. We do have some papers describing canine visceral leishmaniasis in Brazil. So sleeping outside is a risk factor. The size of the dog, dark fur, short fur, age. Normally, older dogs has more seroprevalence. The presence of organic matter on the backyard, and lower family social income. But we do have these risk factors described in literature, but we don't have one paper that puts this all data together. Normally we have one paper that accounts for one variable but doesn't account for other variables. And I did not found one paper that put together these variables and also search for tick-borne diseases exposure. So this was our main of our paper. So we do have the paper risk factors associated with Leishmania seropositive in Fortaleza City, Brazil, environmental, social, Ehrlichia exposure, and dog individual characteristics. So the highlights of this study was the first study that we use a One Health approach in a logistic regression model for visceral canine leishmaniasis. So we did account that we do have the results that exposure to Ehrlichia, the family, social income, and the characteristics of the dogs are related to positivity with leishmaniasis. Our results, if depending on how you interpret it, show that the risk factors that we will show soon that they are independent. If you control for one, the other will still be a risk factor. And this is very important because we need to account for the epidemiology of visceral leishmaniasis regardless of the species. My dream was to account on this same data for more species, but I had to focus on dogs. I cannot collect human blood to test that out because I'm a veterinarian. So, but the true big research work would be collect human blood from the household, dog's blood. If you have a cat blood. It doesn't make sense to include birds, but it can include other animals if it does have on the house, like rats or other rodents or other animals that has on the house. So our objective on this study was to analyze risk factors associated with leishmania positivity in dogs in Fortaleza. We include several variables, as I said to you. So we put variables associated with the dog, variables associated with Ehrlichia positivity, socioeconomic status, and environmental factors. And we use a regression model, multiple regression model. So we included two areas of Fortaleza. For those who live in Fortaleza, was Barra do Ceará, Regional 1, and Conjunto Palmeiras, na Regional 6. And then we calculated the sample size for a cohort. The cohort we did not finish, but because of the pandemic, but we did collect 492 animals on the cohort, the sample size, and then we tested the dogs for the leishmania by DPP. The DPP is the immunochromatograph.
Slide is not passing.
Are you seeing slide 24?
The freezing on the title of your paper. 20. There is slide 20. You maybe get off and get in again.
Deu certo agora ou não?
No.
Okay, let me try to.
Just stop the share and restart. Yeah. Thank you. Yeah.
Okay.
And go to the 20 slide and you talk about the design, I guess.
Okay. So you were all on this, right?
No, not yet. We're not seeing anything.
Why is that? Here it shows me that it's sharing.
It's sharing, but a black screen.
Restart. Close and
Eu vou fechar aqui o PowerPoint então.
Yes, sir.
E vou abrir de novo o PowerPoint. Espera aí. Aí vocês conseguem ver pelo menos a tela do meeting aqui?
Yeah. Sim. No.
O que é a tela do meeting?
Pronto, agora eu parei de compartilhar.
Agora eu estou vendo você.
Pronto, deixa eu tentar abrir aqui de novo. Interessante. Talvez seja a internet aqui do hotel que tenha dado uma caída, não?
Mas a gente está vendo você.
Estão ouvindo, né, também. Ok. Deixa eu compartilhar novamente. Okay, so for me it's sharing. Do you see my shared screen? I'm sharing the meeting.
Vocês estão vendo? Não, tá preto, a tela tá preta.
Tá. Deixa eu fechar aqui. Deixa eu fechar esse aqui. E agora aparece o slide, não?
Não. Adam, é melhor você sair do Meet e entrar de novo.
Entrar de novo, né? Ok, vou sair aqui rapidão.
É difícil.
Ok, vou tentar aqui compartilhar só a janela do PowerPoint. Deixa eu ver aqui. Pronto. Apareceu agora, não?
Sim, agora sim.
Pronto, então tá certo. Eu não vou nem dar um alt tab aqui, não. Então, pronto. Então, is still there, the presentation?
Sim. Full screen now.
Okay.
Você está no slide 20. Yeah, okay.
Yes, okay. So I was talking these slides, they are all texts. Do you need me to repeat it? But so the highlights of the study was to do a unique logistic regression model that we account for diverse variables on diverse areas of variables. So we did account for exposure to Ehrlichia, the family status, and the dog characteristics related to Leishmania. So we did 2 regionals, two neighborhoods in Fortaleza. We did a sample size calculation for a cohort, but the cohort doesn't work it out because of the pandemic. So we did transversional cross-sectional study. We tested the dog by the DPP. If somebody doesn't know, the DPP is a fast immunochromatographic test for Leishmania for dogs used by the Ministry of Health in Brazil. We did tests for SNAP IDEX for Leishmania as well. It's another point of care test. We did test the dogs for Ehrlichia by IDEX SNAP test. It's a 4DX product. And we did the other data set by an inquiry. So we did inquiry with 58 questions with socioeconomic data, dog characteristics, and environmental factors. So the results were described in frequencies. We did analysis on the R program. We did the logistic regression model. And the socioeconomic status was made by a BEPI institution. So it does classify the family from 1 to 7, being 1 the richest and the 7 the poorest. It would be wrong to say just the rich and the poorest because it does account for other measurements as a level of education and other variables. On this questionnaire itself has approximately 30 inputs, and then it goes out from a score from 1 to 7. So it's very fascinating, this method. I did like it a lot. So it's a numerical variable. It goes from 1 to 7, being 1 the less exposed and the 7 the most exposed. The positivity for leishmania, we use and or. So if the dog were positive in one of the 2 tests made or in both of the tests we used, we did not use the only the 2 positive dogs. We used one or other, and you use significance. Tem alguém perguntando alguma coisa? Or somebody asking something?
I'm still not passing the slide. Have you the full screen? All right, now now it's passing. Go for the full screen. I mean,
okay.
Yeah, now it's passing. Yeah.
Okay.
Full screen.
So slide 26. Is it okay?
Yeah, but just put full screen. It's passing slow. I don't know why. Passando.
Okay.
Tá. PowerPoint, manda a tela cheia.
Tá na tela cheia já aqui para mim.
Aqui não tá ainda não.
Não tá, engraçado.
Eu posso tentar. Tá, talvez seja o computador aqui. Vocês estão no slide 26, pelo menos? Vendo?
Tá vendo, é melhor deixar assim antes de...
Pronto. Ok.
26,
Pronto. Então, we did collect a total of 690 dogs. We did include 521, a total of 354 houses. We do have a lot of data catalog. We do have 190 complete blood count made. So we do have a lot of samples to test with a lamp from Rubens if he wants. We do have serum as well, so we can test for serum or total blood. And we do have 196 dogs with 2 points of observation. This would be the first, the beginning of the cohort, but we do have okay, so for our results, we do have, as I said, 521 dogs, a total percentage of 20% leishmania positivity on any test, a 10% positivity on DPP, and a 14% positivity on the SNAP. For Ehrlichia seropositivity, we have 62 seropositivity by SNAP. The interesting fact here is when you have a dog positive for leishmaniasis in any test, it goes the seropositivity for Ehrlichia for 75%. So showing off that comorbidity is somehow related on these dogs, on these 2 species of pathogens. So for household sociostratum, you can see a trend to the most lower income houses has the most the higher frequency both for leishmaniasis and for ehrlichiosis. And we did expect that, but for me it's very interesting to see the numbers so you can really see what's happening. So for the dog characteristic, we did notice higher positivity in dogs with short fur, and bigger dogs. And this is an interesting fact here for bigger dogs and dogs for sleeps outside. So we have 27% of the dogs seropositive for leishmaniasis when they sleep outside. When they sleep inside, it drops to 13%. And for dog size, it goes for 30% in larger dogs and 19% on short dogs, not short dogs, cães menores. This is interesting because when you control this variable for sleeping location, because many of scientists says that the dogs that are larger sleeps outside, but this is not what causes the higher leishmaniasis, because when you see the numbers, it doesn't, when you test it mathematically, it doesn't prove that. So actually, when you see the proportion of dog that sleeps outside, large and not large, the sleeping location, it kind of has the same trend of distribution on the normal curve. So we speculate that larger dogs has more phlebotomy attractiveness to get more leishmaniasis. And this is kind of interesting. This is kind of sitting low on the drawer to investigate further, but it's very interesting to speculate or hypothesize what's happening here. The other thing is we did compare the DPP by SNAP. I don't know if you're interested, but we have a low kappa agreement between the two tests. These are both serological, so it's leishmania. It's easy to diagnosticate, but it's not easy to trust only one method. You need 2 or 3 methods to diagnose these diseases. This is a very tricky disease to detect. For our multiple regression model, we can see here that house sociostratum has a significant odds ratio. So this is esse é o chato de pronto. So this is for each jumping house sociostratum. So 1 to 2, 2 to 3, until 7. So each jump of house sociostratum, you have 1.4 times more likely to have a dog infected or seropositive for leishmaniasis. When you have a Ehrlichia seropositivity dog, you have a 2 times more chance to have a dog seropositive for leishmaniasis. When you have a dog that sleep outside at 2.3, and you have not larger dogs are more successful by 1.8 chances. So it's interesting because you can put sleeping location. Okay, so I will say that on the conclusion. So what we can say is by this study, by this data, is that we can, and this is interesting because when you talk about One Health and you talk about getting families more education and more money or with more jobs, get people better jobs, get people better houses, better education, they kind of trend to not getting diseases. And this is not only with leishmaniasis, this is actually with canine leishmaniasis in my data. But when you go to read about it, you can read about it on HIV, you can read about it on human leishmaniasis, and you can read about it on more diseases. So this is interesting because you could use education and getting people's jobs or better jobs for a health preventive measure. You can stimulate dogs to sleep inside the house. This is kind of interesting. So the dogs that sleep inside are very much protected for leishmaniasis and for ehrlichiosis. Obviously, for not accounting for ehrlichiosis, you can avoid tick contact. So it's easier to avoid tick contact, to avoid phlebotomine contact, because you cannot see the phlebotomine if you're not looking for it. And the tick is pretty easy to control. We have several new drugs today that can be a tool to prevent ticks. So you could use those tools to prevent ehrlichiosis and, as a consequence, prevent canine leishmaniosis. So these could be measurements for canine leishmaniosis. The other thing is that our model showed the importance of the variables studied that I showed to you on the result slides, and canine visceral leishmaniasis are associated with social factors. And we do prospect that the One Health concept or the One Health approach is adequate on urban areas for endemic for canine visceral leishmaniasis. So we do believe that the knowledge of the veterinarian determines the success on the prevention and detection of ehrlichiosis and leishmaniosis. And we reinforced that the One Health approach or concept. It's the concept that would be better for human health, animal, and environment. And if you don't account for these three together, we will be losing energy, time, and resources. So they are related, and we should account for them together. So this is my conclusion. And I thank you very much on everybody that helped me, everybody from the field, my family, my dogs, and I do thank you, and I am open for discussion.
So Anastasia, take over, yeah.
Presentation, and I think Adam is doing a very nice work, and I have to read your papers. I haven't done it, so I hope you come with very good results, so can help this to control this endemic disease. It's a very, very serious problem.
Yes.
Congratulations.
Thank you, Anastasia.
Let me ask one little question, if I can, Anastasia. It's the same question that I did for Anastasia. For me, it seems that for co-infection is more common in dogs than co-infection in human disease. In the primary area, I mean, in the field or clinical outcome clinical. Do you think it's true for dogs? It's more common in dog than co-infection than human?
Yes, we. I can say that as dogs are not like the tick prevention on dogs are not well done, so it's easy to prevent HIV. Maybe because I don't know, because I'm going to take that back because I'm not a doctor and I don't work with people with HIV. But I do believe that to prevent a disease that is transmitted by sexual transmitted or by blood, it's much more slower than a transmission that is doing by ticks, and people are not accounting for tick prevention. We don't have any federal resource to tick prevention. Actually, Anastasia mentioned the collars, and in Fortaleza, the collars are being distributed as a preventive measure for leishmaniasis, and it's a good preventive measure. It's not a 100% preventive measure, but it does slow the leishmania incidence on canines. What we do know is that actually, it's my hypothesis, the canines that has other exposure or other disease, they acquire leishmania easily, but I don't believe that it's because they go together. It's because the dog, I think, what I do believe is the dogs are exposure, and the healthy dogs resolve the problem with lower parasite bites or inoculation. When you have a dog that already have a heliciosis in the subclinical stage or backwards, are dogs that have a leishmania in the subclinical stage. When it has a inoculation of Ehrlichia, inoculation of leishmania, it's more likely to not kill the parasite or kill the bacteria and then become co-infected. So I think it's from there. For sure, if you're a clinic, the dog that have comorbidity has a worse prognostic, for sure.
Thank you.
The problem with HIV is a disease of behavior. It's very difficult to change behavior of the people. So even though we know a lot about the disease, how it is transmitted, new infections are still happening every day, because to change behavior of the people is very difficult. It's very difficult. Those things that we can do with the dog, we can do it. Of course, the environment of the dog you cannot change, but both questions are very difficult to do. It's just amazing how people do not follow what they know. They just follow their instinct. So,
and this is interesting, Dr. Anastasia, because my data show that if you don't go for collars, don't go for vaccine, go for education and better human resources. And this would prevent, like this would, in my data, would decrease leishmaniasis for 1.4 chances by each class. So if you go from 1 to 7, it would be a huge prevention method. So if I could bet on something, I would bet on education and bet on better resources, better salaries, better jobs. It would prevent a lot of disease. And I do believe that this is true in HIV and other diseases, that better education would change the behavior. But
of course, no doubt about that. But that's the point. I think we have to use all the knowledge to put them together and see how can we improve all these aspects of transmission and control the disease in a better way. So
yes.
Congratulations.
Well, I think because of the time, I think we need to close. And then just to remind you that tomorrow we're going to start the section is almost the same time as today, 8:30 in the morning. And it's going to be talking about the SARS-CoV-2 and influenza A and B virus in human and animal stuff. So I think it's going to be a nice section too. So I congratulate all the speaker. It was very nice to hear you and also the assistants, and was very nice. I mean, we did get between 17 to 42 people participate actively in these meetings. So it's the first time that I'm seeing a very good participation, discussion, international discussion too, and it was nice. Thank you very much and have a good night. I'll see you tomorrow.
See you all tomorrow.
Recording has stopped.